डॉक्टर मुरली मनोहर जोशी।
भगवान श्री कृष्ण के समकालीन महाराजा अग्रसेन समरसता और आर्थिक विकास के प्रतीक हैं। उनके मूल्य और संस्कार आज भी सामयिक हैं। 1998 में मेट्स की स्थापना दूरदर्शी और समाजसेवियों के एक समूह द्वारा की गई, जो उच्च गुणवत्ता वाली तकनीकी शिक्षा प्रदान करने के लिए समर्पित थे, जो ईमानदारी, उद्यमशीलता और सामुदायिक सेवा का प्रतीक थे।
दिल्ली सरकार में इंद्रप्रस्थ विश्वविद्यालय बनाने के लिए जब हम लोग कोशिश कर रहे थे तो उस समय कई अफसरों ने कहा कि यूनिवर्सिटी तो आप बना लोगे सर, कॉलेज कौन आएगा आपसे एफिलिएट होने, क्योंकि दिल्ली के अंदर ज़मीन की समस्या है, साधनों की समस्या है, तो बहुत मुश्किल काम रहेगा। तो उस समय हमारे कई मित्रों को भाई एक तो मैं प्रेरित करूंगा समाजों को, तो सिख समाज का काफ़ी योगदान है दिल्ली की शिक्षा में, हमारे समाज का महाराजा अग्रसेन समाज का है, ब्राह्मण समाज, ये 3-4 समाजों को मैं कोशिश करूंगा कि ये अच्छे इंस्टीट्यूट खड़े करें। मेरे मित्रों से बात की और उन मित्रों से बात करके भाई एक संस्था बनाते हैं जिसमें एक इंजीनियरिंग कॉलेज शुरू करें।
आज महाराजा अग्रसेन टेक्निकल एजुकेशन सोसाइटी मेट्स दिल्ली में महाराजा अग्रसेन इंस्टीट्यूट ऑफ टेक्नोलॉजी, महाराजा अग्रसेन इंस्टीट्यूट ऑफ मैनेजमेंट स्टडीज, महाराजा अग्रसेन बिजनेस स्कूल और महाराजा अग्रसेन विश्वविद्यालय बद्दी हिमाचल प्रदेश जैसी संस्थाओं के द्वारा अत्याधुनिक सुविधाओं और व्यावसायिक पाठ्यक्रम के साथ शैक्षिक उत्कृष्टता के एक स्तंभ के रूप में खड़ा है।
डॉक्टर मुरली मनोहर जोशी जी जो उस समय शिक्षा मंत्री थे, उन्होंने जब ये कहा कि मैं बैठा हूं, कोई दिक्कत नहीं आएगी, शुरू तो करो, और हमने शुरू किया। और जब इस नए भवन का उद्घाटन माननीय अटल बिहारी वाजपेयी जी से करवाया तो उस समय लगा कि हमारी मेहनत का कोई पहचान तो बनी। क्यों, अटल बिहारी वाजपेयी जी से पहले मैंने जहाँ ये इंस्टीट्यूट शुरू किया था, गीता स्कूल सुल्तानपुरी, वहाँ हमने रजत जयंती में अटल जी आए थे, और जब उनको ये बताया कि वहीं से शुरू होकर हम यहाँ आए हैं तो ये आगे बढ़ो गर्ग जी, आप में बहुत क्षमता है। अब उनके एक कहने से ही हमने मैनेजमेंट ऑफ इंस्टीट्यूट भी शुरू कर दिया। भगवान की कृपा से वो भी अपना एक स्थान, और उसके अंदर हमारे कई मित्रों ने कहा कि जर्नलिज्म शुरू करो, और हमारे कई बड़े अटल जी ने भी कहा कि अच्छे जर्नलिस्ट, राष्ट्रवादी जर्नलिस्ट आप पैदा कर सकते हो आप उनको, तो हमने जर्नलिज्म का कोर्स शुरू किया। फिर लोगों को अच्छे वकील भी हम देश को दें, अच्छे जजेस दें, तो हमें लगा कि हमें लॉ भी शुरू करना चाहिए। और एक पहाड़ों पे पढ़ने का शौक़ है, उसे देखते हुए हिमाचल में महाराजा अग्रसेन यूनिवर्सिटी, जिसका उद्घाटन आदरणीय राष्ट्रपति प्रणब मुखर्जी ने किया, ये प्रोत्साहन मिलते रहे, आगे बढ़ते रहे।
पिछले कुछ वर्षों में मेट सोसाइटी की संस्थाओं ने कई कीर्तिमान हासिल किए हैं। हजारों योग्य स्नातकों का प्रशिक्षण, तकनीकी शिक्षा में शीर्ष रैंकिंग और अग्रणी उद्योगों के साथ सहयोग भी स्थापित किया है।
महाराजा अग्रसेन इंस्टीट्यूट एनबीए एक्रेडिटेड इंस्टीट्यूट है। इसके 3 प्रोग्राम्स बीटेक, कंप्यूटर साइंस एंड इंजीनियरिंग, मैकेनिकल एंड ऑटोमेशन इंजीनियरिंग, इलेक्ट्रॉनिक्स एंड कम्युनिकेशन इंजीनियरिंग, आईएसओ सर्टिफाइड है। एनआईआरएफ की रैंकिंग है 201 और 300 के बीच में। बैंड प्रॉमिसिंग इसको अटल रैंकिंग के थ्रू मिला हुआ है। 35 एमओयूज हैं इसके, जिसमें कि 3 इंटरनेशनल एमओयूज हैं, और ये ग्लोबली रिकग्नाइज्ड है। रिसर्च बेस्ड हम लोग बहुत सारा काम करते हैं। जो बड़ी बड़ी ऑर्गेनाइजेशंस हैं जैसे एआईसीटी हो गया, डीआरडीओ हो गया, इस तरह की ऑर्गेनाइजेशंस के साथ में हम लोग कोलैबोरेशन भी रखते हैं और इनके साथ बहुत ज़्यादा रिसर्च प्रोजेक्ट्स भी करते हैं।
महाराजा अग्रसेन इंस्टीट्यूट ऑफ मैनेजमेंट स्टडीज़ नाम के पौधे को 2003 में महाराजा अग्रसेन टेक्निकल एजुकेशन सोसाइटी द्वारा रोपा गया। तब से आज तक ट्रस्टीगणों, अध्यापकों और विद्यार्थियों द्वारा इस पौधे को बड़े प्यार से, मेहनत से, निष्ठा से एवं विवेक से सींचा गया, और आज ये पौधा एक वृक्ष का रूप धारण कर चुका है। आज हम नैक A++ हैं और हमारे विद्यार्थी हर क्षेत्र में आगे बढ़ रहे हैं।
महाराजा अग्रसेन संस्था ने ग्रैंड थॉर्टन, जो दुनिया की 6 बड़ी कंपनियों में काउंट होती है, के साथ कोलैबोरेशन किया। उस कोलैबोरेशन के अंतर्गत एक महाराजा अग्रसेन बिजनेस स्कूल को इस्टेब्लिश किया गया। उसके बाद AICTE ने उसको मान्यता भी प्रदान की और अब यहाँ पर 2 साल का प्रोग्राम कंडक्ट किया जा रहा है। ये प्रोग्राम केस स्टडी बेस्ड मैथडोलॉजी से पढ़ाया जाता है। इसमें इंटरनेशनल कोलैबोरेशन होने शुरू हो गए हैं। पिछले साल बच्चे दुबई गए थे, इस साल न्यूजीलैंड की एक बड़ी यूनिवर्सिटी के साथ करार किया गया है। इस तरह से इंटरनेशनल कोलैबोरेशन और केस स्टडी बेस्ड मेथड के साथ में एक इंटरनेशनल लेवल का प्रोग्राम इस संस्था में अब चल रहा है।
अकादमिक शिक्षा से परे मेट्स राष्ट्र प्रथम की भावना के साथ समाज को वापस देने, छात्रों को सामुदायिक सेवा के कार्यों में शामिल करने, सामाजिक जिम्मेदारी और नेतृत्व की भावना पैदा करने में विश्वास करता है।
हमने 2 रोल मॉडल लिए थे, मदन मोहन मालवीय जी का हिंदू यूनिवर्सिटी और महात्मा गांधी द्वारा स्थापित रोहतक में वैश्य इंस्टीट्यूशंस, जिसके अंदर शिक्षा का व्यवसायीकरण ना हो, शिक्षा सामान्य लोगों को उपलब्ध हो और जिसके अंदर राष्ट्रवाद को, नेशन फर्स्ट, यह भावना लेकर चलने वाले नए जो मानव हैं, जो नए शिक्षित लोग हैं, वो हम समाज को दे सकें।
आज जब हम अपनी रजत जयंती यानी 25वीं वर्षगांठ मना रहे हैं, हम उत्कृष्टता की अपनी विरासत पर विचार करते हुए उज्जवल भविष्य की आशा कर रहे हैं।
A very good afternoon, everyone. A warm welcome to the last day of our ATAL Faculty Development Program. We hope you are refreshed and ready for another enriching day filled with insightful sessions and engaging discussion. Let's make this day as productive and exciting as the previous ones. Now, we have an exciting session on neurodegenerative disorders and 3D model analysis by Dr. Rohini P., Assistant Professor in the Department of Electronics and Communication Engineering at the Indian Institute of Information Technology, Design and Manufacturing, Kanchipuram. As a speaker for this faculty development program, Dr. Rohini academic journey began with her PhD in biomedical engineering at the Indian Institute of Technology, Madras, where she joined the NIID laboratory in 2016. Her doctoral research focused on neuroimaging studies to uncover intricate variations in Alzheimer's disease utilizing MRI, image processing, and machine learning techniques. Her thesis delved into identifying textural changes associated with brain-based Alzheimer's traits, demonstrating her expertise in advanced image processing algorithms and brain imaging tools such as BSC, BET, BrainSuits, Neuron, SPM8, and MATLAB. Following her PhD, Dr. Rohini was awarded a pre-doctoral fellowship by IIT Madras to further her research at the NIIT lab. In 2016, she joined Triple ITM Kanchipuram as an assistant professor, where she established the Translational Healthcare Research and Innovation Lab. This lab focuses on medical image analysis for neurodegenerative diseases and biosignal processing projects. Dr. Rohini expertising in neuroimaging, medical image analysis, and translational healthcare innovation promises to provide valuable insight and inspiration to the participant of this faculty development program. We are honored to have you with us, Dr. Rohini, and we are looking forward to your session. And now, before starting the session, a quick reminder for all the participants. Please keep your microphone muted during the session to avoid any kind of interruptions. Use the chat box for any questions or any other comments, and we will address them during the question and answer segments. And please don't forget to fill out the feedback form at the end of this FDP. And now I would like to invite Dr. Rohini to take the stage and share their valuable insight. Over to you, ma'am. Please.
Thank you very much. Thank you for this warm welcome. On the other day, I'm very excited to be present here. And thank you, Dr. Nitin Sharma sir, for having me here. So yesterday we were, I'll just present my screen, then we can start the session. I think it's visible.
Yes, ma'am.
Yeah.
Yes, ma'am, it's visible.
Yeah, thank you very much. Okay, so yesterday we were discussing the basics of image processing, how what kind of techniques are available for us to deal with and to implement it in the analysis part. Today we will go in depth into a case study based scenario. I'll give you 3 case studies. One is based on neurodegenerative disorders and how to extract 3D information from that, and how do we analyze it. This is one part. The second part will be based on another cell imaging technique, which is also analysis with a deep learning methodology. So I have planned it in such a way that you will have a flavor of both image processing, machine learning, and deep learning approach. So to begin with, we need to understand what is a neurodegenerative disorder, right? So we had spoken about so many different imaging techniques, that is starting from X-ray, CT, MRI, and other things. So all these techniques are developed for understanding how the disease progression is, what kind of diseases are affecting the body condition and other things. So when we talk about brain specifically, brain has a major function called to be as cognition. Okay, so what is cognition? Cognition is nothing but how aware are we? So we are thinking, our thinking capability, our perception, our reasoning capability. All these things come under the terminology called to be as cognition. Okay. So it basically represents the brain activity that is happening every single time we are awake, we are conscious. Okay. So cognitive impairment is another terminology that is also related with cognition, wherein it involves problems that are associated with memory. Okay. So when we are having some kind of thought process that is that we are looking at, every time our memory unit gets activated. So our memory unit is comprised of hippocampus. Okay, so that is getting activated and it is storing information in the hippocampal region. Now, an impairment in this cognition process is called to be as cognitive impairment, and it involves some memory problems and thinking deficiencies and other things. So this cognitive impairment, so there are many films, there are many awareness that have been given towards this cognitive impairment. Recently, many films, even in Bollywood, Hollywood, they have been made on these cognitive impairment disease conditions and how they are affecting the daily life of every people who are affected by this disease condition. Okay, so now basically, if we see that it accounts for about 13% of the global disease burden, it's a very huge amount actually, and mainly it is affecting the old age population. We know that very well, but it is not only old age population that is actually in the scenario, but even the people who are above 60 years of age. So we generally don't term them as old age, but still they are also affected due to this cognitive impairments. Okay, what are the factors? There are several factors that are contributing towards this cognitive impairment. Normal aging. So even because of the aging, there are some changes happening in the brain cells which can contribute to these kind of impairment. But the other major reason is some kind of disease condition, some disorders that can lead to this cognitive impairment. Okay, so one major disorder that that leads to this cognitive impairment is dementia or dementic condition. But there are differences between how normal aging is affecting the cognition and how neurodegeneration is affecting cognition. Okay, so those two, that difference is very important because nowadays, if you think about our society, okay, India is a huge, heavily populated nation. And here, even if you think about the old age population, the percentage of old age population in India is actually very high, pretty high. And it is continuously growing based on the improved healthcare systems and other things. It is continuously growing. Okay, now if you by yourself you would have an idea that whenever you take a old age person to a hospital, to a clinic, stating that they are actually forgetting certain things in the old age, they simply evade saying that it is just due to old age, we cannot do anything about it. But it is wrong actually. There are differences that are available when you a normal aging leading to memory deficits and neurodegenerative disorders leading to memory deficits. So if we can identify the difference between these two, we will be able to cure certain of them. I will not say cure, but you can think about stopping the progression of the disease condition. Like it will not further worsen. Okay, so wherever, at whichever point that person is, at that point we can stop the progression or we can delay the progression. So these things are available still in the industry. Okay, so right now we are concentrating on neurodegenerative disorders that are leading to cognitive impairments. What they do is they primarily affect the brain cells. Okay, they are called to be as neurons, and they are the building blocks of the nervous system, which are comprising of brain and spinal cord. So we know this very well. So these are all basics. So now, what happens in the neurodegenerative disorders? So when there are when it is affected by neurodegenerative disorders, what happens is there are damages or death to the neurons that are happening, which are very significant, and these neuronal cells are such a way that they cannot be reproduced or it cannot undergo regeneration. That is why it is called to be as neurodegenerative disorder. So once it is dead, it is dead. That's it. We cannot regenerate. It's not like our skin cells or other muscle cells which can regenerate it by itself. So that is not available in the brain cells. So once it has been matured, so once we attain a certain age, the brain cells are matured, and after that we will not be able to regenerate any lost cells. Okay, so what are some of the examples of these neurodegenerative disorders? So we have Alzheimer's disease, which is very, very predominant. Parkinson's disease is there, and we have frontotemporal disorder, Lewy body dementia. There are different kinds of dementias itself that are available. So, and also all these are named in such a way that it affects different parts of the brain. One, and also the condition because of which the brain cells are getting degenerated, that is different in each condition. Like for example, Alzheimer's disease, we have different theories that are causing Alzheimer's disease. One say neurofibrillary tangles are present in the. Let's not go into details of it, but still, whatever the differences between the disorders are different. So only if we identify, why I'm emphasizing on this is only if we identify what kind of disorder it is. Like for example, if it is Alzheimer's or if it is Parkinson's, all have a common symptom like memory deficits. Okay, everything affects memory. So memory problems are there, problems are available. So then we cannot conclude that the person is having Alzheimer's. Not possible. So we have other dementias also that affects the brain condition. So that is why differentiating between Alzheimer's, Parkinson's, frontotemporal. So all these, there are different, there are about 10 to 12 types of degenerative disorders. So it is also essential to have differences between each one of them. Okay. Now, to start with this cognitive impairments, we will take an example of Alzheimer's disease. Okay, we will focus on Alzheimer's disease so that it becomes narrowed down. We can focus on one thing. We can see how we can analyze it. What types of analysis can be done. Now, so if you can see the image here, which is given, you can see a normal brain, how intact it is, how the brain cells are located, and what are the so these are all called to be as this is a cortical region and this is the cerebellar region. Now, you can see how intactly the brain cells are present. Okay, whereas in the AD, that is Alzheimer's disease subject, you can see this is a brain image of an Alzheimer's disease subject. You can see there are so many brain neuronal loss that has happened because of which the brain cells are having some gaps, right? So the tissue region is having gaps, which are actually eventually it will be filled with CSF fluid because it cannot remain as a gap, right? Inside the body, there will be CSF fluid that is present, which will fill these gaps. But you can see the difference between the normal and the AD. So this is a 3D representation of a brain. Okay, it's an MRI actually. So 3D representation, you can see it as an object. Like now, the same thing if I do a slice here, if I take a slice in the middle, and if I view it, okay, so this is a 3D slice of the normal brain, okay, and if you can see the region of the brain that is very intact is having healthy neurons that are present, and here you can see the AD, the similar 2D view of the AD is also seen. You can see there are so much of gaps that are present in the between the tissue cells, and that is why we have loosely packed tissue cells that are present in the AD. This is mainly caused due to degeneration. Cells are dying, and the space is actually getting filled with AD. But sorry, space is getting filled with CSF. Okay. Now, a person does not directly get AD overnight. It is not possible. So everything has a development stage. Okay. So how the development is actually happening? So it starts from a normal person turning into getting something called to be as early mild cognitive impairment. So cognitive impairment we were talking. So there is something called to be as mild cognitive impairment and early mild cognitive impairment. So these stages are actually given by a staging system which is called to be as Brack. Now, this staging system is actually a standard staging system that is being followed in the local community, right? So, like for example, if you go to a doctor, based on the certain test, they will assess you and they will fit you into one of the stages. So, when we start with early mild cognitive impairment, it takes time to progress towards mild cognitive impairment, like about a year or two. Okay, and from here to late mild cognitive impairment, another 2 years or 3 years, depending on how the subject is. Okay, and finally only it will go towards Alzheimer's disease, the final stage of the disease condition. So if you can see this timeline, it is actually quite a significant timeline. So it is not happening overnight like other disease conditions, like for example, other heart-related disease conditions or whatever it is that we are looking at in a very immediate stage. That is not like it is. Any neurodegenerative disorder is not like that. It takes time for the progression. So when we can identify this disease condition in an early stage, very, very early stage, that is early mild cognitive impairment, which is called to be as a preclinical stage or asymptomatic stage of the disease condition. So what is this preclinical stage? Before the symptoms are being visible. Okay, so for example, I told you that a person with cognitive impairment can lead to AD, right? So cognitive impairment means I know that the person is having some kind of memory issues, correct? So, but without even having memory issues before this stage, can we identify can we predict whether the person is going to get Alzheimer's or not? So that is what is this EMCI or early mild cognitive impairment stage. Wherein if we can identify this stage, we can very well stop the progression to the Alzheimer's disease. It's highly, it's a very a sensitive stage wherein if we can take prior notice and identify it in a very effective way, then it is highly possible to stop the progression. So that is what is our main aim. What are the techniques that are generally used to assess this early mild cognitive impairment? We have different imaging techniques, but you think one information that I don't have any symptom, right? So I don't have any memory deficits or anything. A person or an old age person who is not having any memory deficits or anything, why will he generally go take a test? Okay, but this study is a controlled environmental study. This can be translated into a study which can be used in the clinical practice. Okay, so that is why we are taking different kinds of imaging techniques and seeing where it is highly visible. So which technique is highly suitable. Okay, so there are different imaging techniques that are being used to assess the cognitive impairment. We start with PET. PET is a positron emission tomography. Amyloid PET specifically is there. So this amyloid PET, amyloid is a kind of a protein that is actually present in excess during the Alzheimer's disease condition. Okay, so we generally measure the amyloid pathology in the using PET images. So that is why it is called to be as amyloid PET. FDG PET is we measure the glucose levels. Okay, in the that is being taken up by the glucose levels that is being taken up by the brain. So this PET and this PET is different, and we have MRI. So structural MRI, normal structural MRIs are there. We have functional MRIs. We can take DTI measures. So all these techniques have been used to measure to assess the cognitive impairment. But generally, we don't directly go into scans. I told you this, right? So we take subjective measures primarily. So subjective assessment is like assessing the cognitive state of a person. Okay, so how well he's responding to the questions that are being asked. So in the subjective assessment, we have something called to be as MMSE or mini mental state examination. So using this, the doctors try to put you into one category, wherein whether you are normal, whether you are facing any issues or something like that based on the test that they will do. Okay, so they are like subjective scores, subjective tests that are being done. And if they want further test, if they are finding that you are having some kind of problem with the cognition, then they ask you for going for the further test. Okay, so this is the initial assessment, initial screening, I can say, that we do for taking for understanding the cognitive state of a person. Now, okay, so after among all these, let us focus on one thing that is called MRI, or we will call it to be a structural MRI to understand, to emphasize on the fact that it is actually capturing the structural changes in the brain. Like structural changes means, I told you that there are loss of neuronal tissue. So the loss of neuronal tissue will actually reduce the integrity of the between the cells, right? So the connectivity, integrity between the cells are being affected. So what we try to do is we are we are going to understand how the structural changes are happening within the brain cells, and that can be captured using MRI, and how it is affecting how it is related to cognitive impairment. Okay. Now, so to understand the basics, because we are going into depth with MRI, so I want you to understand what is actually how to read an MRI, so that when I show you some images which we have actually processed, it will be easier for you to understand. So a raw MR image is like this. So we will be using three different dimensions to record MRI or to view MRI. Okay, so I will not say record. Recording is happening only in one axis, okay, one dimension, and we will be interpolating it to view in different axes. Like for example, you can think about an object, okay, and you're rotating the object and seeing in different dimensions, right? So it is just like that. So this is the a three-dimensional image. So you can see the axial view. Axial view is nothing but if you see from the top. Okay, so from the top, how your brain is. So that is what is shown here, and it is just one slice that is being represented. Okay, so this is coronal view. Coronal view is from the back or front to back view. Okay, so from the front or from the back, how when you see the brain, how it is looking like. So this is the brain region. Okay, this is the brain region. And from the sagittal view, when you see it from the side, how the brain is looking. So you can see apart from the brain tissue. So this is the brain tissue that is there in the center. You can see here, this is the brain tissue. And you have, apart from the brain tissues, you can, when you're doing an MRI, you will have other regions also that are being imaged. You can see nose region is there. You can see the eyes, eyeball is there. Okay, and this is a skull region. Skull is actually not visible. Rather, we can say meninges and dura, the outer covering, the tissues that are outer covering of the skull. So that is actually visible. And dura is there, the outer covering of the brain tissue. Okay, and we have different parts of the brain. This is the cortical region. So the top of the brain is called to be as cortical region, cortex, and this is a cerebellum. This is brainstem. We have multiple. This is an empty space which is called to be as CSF. It's a fluid-filled space, empty space, which is called to be as ventricles. Okay, and this is corpus callosum. If you can see this. this C-shaped structure. This is called to be as corpus callosum. So this corpus callosum, as you know, our brain is made up of two hemispheres. Okay, so you can think about two different balls that are there, and these two balls are connected using a tissue layer that is called to be as corpus callosum. This is what is corpus callosum. Okay, now when you understand the different views of MR image, let us go into what are the problems that we can face when we are working with MR images. First thing, you take different subjects. Okay, I have given you some examples, one example in each. Okay, so I have taken different subjects. You can see each subject is having kind of variation in shape. Everybody's brain is different, everybody's head size is different, so everybody will have variation in the shape in the when they are recording the MRI. Okay, the second one is orientation. So I can keep my head like this straight. I can tilt my head in either way when we are recording the MRI. Okay, though, I mean, if you had taken MRI or if you had seen others taking an MRI, you know that we have something called to be as head cap, where there is particular place in the MRI scanner where you place the head, and it will try to reduce the movement of the head and make sure that it is in the same direction. But still, even if you do have all those things, even then you will have some differences that are arising in the head orientation. Okay, now you can see among the 5 images that are being shown here, every image has a different orientation of the head. So that is also a problem. Okay. The third one is I am interested only in analyzing the brain tissues. Brain tissues are my interest. But as you can see, you have different kinds of tissues that are available, like other than brain tissue. Like for example, the extracranial structures in the skull, the nose bone, nose areas, the eyes, and the mouth region, the jaw region, everything is visible, neck, everything is visible in the MRI. So if we are giving the entire image for analysis, it will create a problem. Okay. So you use any technique, machine learning, deep learning, image processing, whatever it is, if you give the entire region, like the entire image as an input, then it is going to create a problem in the analysis, because the influence of the other structures are also having an important role. Okay. So now, other than this, what is happening? So you can see here, I have differences in the image contrast. So every person has differences in the tissue intensity levels, or I can say the composition levels of the soft tissues, right? So every person is different. They have completely different what is it, composition and other things in the brain. Though we have a common structural composition, the functionality and the nature of the connective tissues, everything will be slightly different for each person. So that will have a difference in the image contrast. So that is also different. And also we have multiple, if you can see the image here, the brain structure or the brain tissue here, you can see there are multiple regions. That is, cortical region is there, corpus callosum is there, brain stem region is there, cerebellum region is there. So there are many different regions that are embedded within one MRI image, that can also create problems when doing the analysis. Okay, so what is the solution for all these things? So when we are doing any MRI based analysis for anything, like for example, for deep learning application or for image processing application, machine learning application, we have to follow certain things that can enhance our results, enhance our accuracy. The first step you do is registration. So I told you that there are differences in the orientation, size, and shape, right? So these are the differences that are available. So first, we generally need to register the image. So what is meant by registration? You can think about matching. Okay. So I have an atlas. I have an atlas means an image that contains regions where I say that, okay, at this point, we need to have what is it? Corpus callosum. At this point, it should be having cortex. So like that, we have an atlas. It's like your Earth's atlas, right? So we can think of Earth atlas, and we know for sure that, okay, at this point, this particular country is there. So it is like that for brain also. We have different atlases that have been created. Okay, so now we need to match our image to that atlas. So what are the techniques that are available? Many techniques are there. Okay, so I'm not going into depth with these registration. I'll give you some ideas what is there. You can actually go in depth and understand what kind of how you can perform that. Okay, so now, image to image registration is there, image to model registration is there, model to model registration is there. So we also call this registration to be a spatial normalization. Spatially, I'm normalizing the regions so that every region, okay, so one particular region of the person's scan corresponds to the same location in the other person's scan. So for example, if I say eyes are here in this particular scan, means the same eyes will be present in the other person's scan in the same location. Okay, their eyes will be present in the same location. So that is called to be as registration. So what are the general available templates that can be used for registration? So we have different brain templates, global brain templates. Which is called to be as Talerac Atlas and MNI templates that are available. Okay, but so we can use these templates to register our brain to the actual atlas using any of these techniques. So I will just show you the registration process. So we can also have one more thing. In this registration, we can also have other process. Like for example, there can be unimodal registration and multimodal registration. What is this unimodal registration? Modal means a type of scan. Okay, one type of scan can be registered, or multimodal, different scans can be registered. Meaning different scans, meaning I have MRI, I have fMRI, I have DTI. So a person's a scan, like for example, I take 3 different scan for a person. Okay, and I can do a registration of these 3 different scans so that I can have multiple information from multiple areas, from multiple scans that can be combined into one image. So that is called to be as unimodal registration and multimodal registration. Okay, now the next process after doing a registration, I'll show you an example in a while. Okay, so the next process in in the pre-processing of any MRI images, we call to be a skull stripping. Okay, why is this skull stripping? I told you we are only interested in the brain tissues. Okay, we are not interested in any non-brain tissues structures, that is skull, eyeballs, bone, all these things we are not interested in, right? So skin and other other dura and other things. We are not interested in any of the other structures. So what we generally do is we have different algorithms that can remove the non-brain tissues. Okay, so that process is called to be a skull stripping. This is one of the algorithm that I have demonstrated, which is actually an active region growing based method. Okay, so we can obviously use deep learning for that. So you can take deep learning models, you can train them for performing skull stripping. That is perfectly possible. Okay. Now what we have done here is, you can see the inside of the brain region is given as a seed point and it is being grown, and you can see only the brain tissue is being segmented. All the other regions, that is the outer region, that the skull, is being removed off, right? Okay, so now this is just an example of how we have done it on our images. You can see normal image, EMCI and AD. I have just given 3 sets of example. Okay, now these are the raw images. You can see the orientation and the size variations that are there. Now if I register it to the spatial to some atlas like Talerac Atlas, MNI Atlas, something. So what happens is, you can see the scan is exactly fitted into the one template. You can see every scan has certain regions that are defined. You can see what is that? Brainstem is present in this region only, in the towards the end of the image, towards the lower end of the image. The corpus callosum is present in this region only. The cortical structures are present on top. So you can see it. It's very clearly it has been localized towards the within the image. Okay, and the next part that we were focusing on is skull stripping, right? So if I take this image and if I strip out the regions which are not needed, like for example skull regions, and this the other extracranial tissues here that is present near the nose and other things, so those tissues will be removed. So you can see the skull stripping has been carried out, and the results are you will get is like this. This is a sagittal view. Sagittal view means from the side. Okay. So I'm showing only sagittal view here. The other view I will show in the next slide. Okay. And the next thing is contrast enhancement. So we have covered this in the last lecture, so I'm not going into the depth with this. So I told you the image contrast will be different, right? Because of the innate differences between the person's brain structure. So there are differences that can happen within the image contrast. So when we enhance the contrast of the images, the visibility of the images will also be enhanced, and this will aid in better accuracy with the algorithm. So we also contrast enhance them. So you can see here, I have also given some of the examples for I have also given some of the examples for. Am I audible? Sorry.
Yes, ma'am, you are audible.
Yes. Okay, fine. Sorry, there was a power cut, so I thought there will be a. Okay, just a minute. Okay, so I am also giving you the representative images for different angle, different view. Okay, so these are all axial views. Okay, so raw images of axial views. You can see here how the axial view images are represented and how the pre-processed images are, the skull stripped and the registered image. Okay, registered and skull stripped image. How is it looking like? So here also these are one the other pre-processed. In the sagittal view, I'm showing you other pre-processed image. So this is also one type of registration and
Ma'am, you are not audible. I think some connection issue has developed. Nitin sir, can you call ma'am and inform her?
I think some technical glitch is there. Wait, and we will contact with the ma'am.
Rohini ma'am was saying that there is a power cut at her place. She was mentioning it.
Then we can arrange during this time other speaker, which
You may first call ma'am and ask
no,
her
no, no.
what
I
is
am
the exact.
here. I have joined back.
okay, ma'am.
Sorry, thank you. Sorry for that. I had just a power cut because of which my internet got switched off. So yeah, we can continue. And okay, so these are the pre-processed images. So now we are interested only in a particular region of the brain, right? So what we have to do is segment that particular region. I am talking about 3D segmentation here. It's not just plain 2D segmentation, not just one slice. Okay, it's a complicated thing. In the sense that an object, if you think about a 3D object, Right now, everybody is focusing on 2D segmentation analysis. For brain, it's information. Like I have a certain each item contains a 3D information. Different slices are there. So what we generally do is there are different approaches for segmentation. We will first see about a normal approach with image processing. So we can do pixel-based segmentation, region-based methods, edge-based methods, clustering-based methods. So this is one thing. Active contours. I had indicated it in the last class where we use region growing methods to segment the particular region of the brain. This is one of the active contour method, but I would focus on one thing which is called to be as a brainstem segmentation technique, which we had done. Okay, so in this we have initiated a novel method. We have proposed a novel method. What the novel method is based on integration of two different methods. It's like an ensemble method you can think of. So that includes clustering with the help of level sets or active contours, wherein what is happening here is we have the brain regions that are being clustered, right? So like for example, we have gray matter, white matter, and cerebrospinal fluid. So these gray matter, white matter has a certain characteristics where the subcortical regions or the white matter have gray level intensity values of very high, like white towards 255. I can you can think about in that way. So you can see the brainstem, the corpus callosum, and the cerebellar regions are highly visible in this image. Okay, so these three regions are being clustered, and it is being extracted first. And by using active contour techniques, you can actually segment out the brainstem region. So like this, you can do on multiple slices. Okay, so this is one slice which I am showing. This is actually sagittal slice. So like this, the entire brain region can be covered, okay, for all the slices. And you can see this is a 3D segmented brainstem structure. Okay, so it is one side view is shown here. You can tilt it and see it will be actually having a 3D structure. Okay, so this is a brainstem that is being segmented as a 3D structure, and we can do analysis on this brainstem region for the further understanding. Like this, we have also used, we have also segmented other regions, which is one is called to be as a ventricular region. You can see this black region in the center, right? So that is a empty space. I had told you, empty space filled with fluid, that is cerebrospinal fluid. So that region is also segmented. So we want to analyze three things. One is brainstem, corpus callosum, and lateral ventricles, and show its effect on early mild cognitive impairment state. So that is what we are trying to do here. I am just trying to explain how we have done that here. So now these ventricular regions are there. So you can see the differences in the ventricular regions. As the disease progresses, the ventricles are bulging. Okay, they are actually growing in size. So this is not, and one more thing, this is not for the same patient. It's not a longitudinal study. It's a cross-sectional study. Okay, so this is for different patients. And also the other segmented corpus callosum is visible here. So these are all 3D structures that I'm mentioning it to you. So 3D corpus callosum is also shown here. So you can see the corpus callosum is having a C-shaped structure. Okay, in all the cases, it is having C-shaped structure wherein you can see the region as similar, that is because of the spatial normalization. So wherever it is present, it is in the same place. Okay, and so after this, what are we trying to do? So we have also we are going to do two things. One is called to be as 2D analysis and 3D analysis. Okay, 2D analysis means I am going to select a slice which is important in that particular 3D structure. Okay, slice means a 2D image. I'm going to select a 2D image which is important as per the literature, as per our method, and then we are going to do the next thing. Now, one more thing is whenever we do any segmentation technique or anything, for example, you take any methodology that is there in your in the process of segmentation, we need to validate them. Okay, so how are you going to validate any segmentation process? Now we need to have a gold standard for validation. Gold standard means like somebody has done that, some doctor or somebody has approved that this is what is the segmented area. So that we need to gather. Now what challenge we faced is we had thousands of images. So no doctor is free enough to sit and do for thousands of images. So what did we do is we had a software that is actually a gold that is actually a gold standard, which is being used by neuroscientists, which is called to be as free surfer software. And this using this free surfer software, what we try to do is segment out the ventricles and compare it with our algorithm. So it will give you some results which by which we can validate whether whatever method we have submitted one thing that I will say is free surfer is actually image, like the say one image takes 10 hours, 11 hours you can imagine. Okay. So if I have thousands of images, I will easily take a month. Okay. So affording that much in-house technique for other things I have shown you for these lateral ventricles, corpus callosum and brainstem. So our own in-house technique is there. I will probably share the paper with you later on. I'm not going to discuss about it because it takes it's a complicated network. Which is a novel network which we created just for using it through the process for segmentation. Okay, so now right now what I've shown you is actually a comparison between the free surfer and the in-house methodology, which is actually a level set method, a level set based technique, which is fuzzy clustering based level set technique. Okay, and only one slice is shown here. Now, what after segmentation, what are we supposed to do? We are supposed to extract features and we are supposed to see how the variations are there. So the basic features we can extract. Okay. Basic volumetric feature you can see across the stages. We can see there are not much differences in the volume. Okay, this is for brainstem, whatever I'm showing you. But for the other regions, you may find variation, but not in the earlier stage. Earlier stage, we are not having any variations in the volume. Okay, when we check out the texture measures, texture features. Okay, there are different kinds of texture measurements. So we can see that there are some variations that are happening, but there are also a huge overlap within the variations that are there across the disease conditions. Okay, so now we can consider texture to be as a good feature to do further analysis. And here I have also and how this local biology pattern is varying across the different brainstem regions. Okay, so now you can see I have taken 3 different classes to present EMC and AD. So here the blue region represents normal texture. Yellow region represents some inhomogeneity is there in that texture. Okay, heterogeneity or inhomogeneity you can call. So homogeneous texture and heterogeneous texture. So you can see as the disease condition progresses, okay, I can see there are more yellow patterns that are getting to appear. More yellow patterns are here. So you can see here also more yellow patterns are there. So this indicates that as the disease progresses, okay, even though from normal to EMCI, if I progress the disease condition, you can see the variation in the yellow patterns. Can you see the yellow pattern is beginning to appear over the edges, and it is slowly spreading towards the interior. So when a person becomes fully having a fully developed AD condition, the entire brainstem is actually having a heterogeneous pattern. Okay, so that was what we had identified, and this is mainly in due to the disease condition due to the there are many different theories for why this is happening. One theory I had told you, neurofibrillary tangles. The second theory is amyloid plaques that are being developed. There is no conclusive evidence that either which is better, which is actually true. Okay, still people are doing research on understanding the theoretical aspect of the Alzheimer's condition, but we can right now from our research we can conclude that brainstem is actually having a difference in effect in the early mild cognitive impairment stage, which can be further enhanced and which can be further analyzed. Okay, so the other this is actually texture based measures, whatever I had spoken. There are other extraction techniques that we can do. One more thing which I want to emphasize here is, see, whenever you are using any deep learning techniques or anything for analysis, I would suggest you to take till segmentation stage, then go forward for the analysis. Because if you directly give any a complete image to the deep learning model, it will surely have an effect on the remaining brain tissues which are not actually involved in the process of analysis. So I would suggest you to take the segmentation process. If necessary, you extract the features also, well and good, extract the features also and give it to a deep learning model, so it will have more precise understanding of what you actually want to do. Okay, now the next thing that I am interested in is shape features. So shape signatures are actually identified. So what is a shape signature? Shape-based methods. Now you can see that I have a ventricular region. This is ventricle, right? So in a 3D structure, if you think about the 3D structure of the ventricle, it is a 3D object. Slice by slice, I am taking the 3D object and I am adding all the ventricular structures. So I will get the shape of the ventricle. This, you can see, it is just the shape, how the edge information is, how the shape of the ventricle is. So with the shape, I am going to do further analysis. This is also one more thing that we had tried. Okay, and similar to this is corpus callosum based technique. So corpus callosum also we had extracted slice by slice, and we have a corpus callosum shape, and we are going to do analysis with this corpus callosum. Okay, so ventricles and corpus callosum shape is being captured, and we are going to perform some analysis. So, and the next thing is what we try to do is to capture the shape signature. So what we did is, you can see the red dot here. We tried to calculate the distance between this red dot. This is the centroid of the image, okay, centroid of the object. And we tried to capture the distance between the centroid and the regions of the edges. Okay, so, and we tried to start from one point and go through the entire 360 degrees. So like this, the same thing is being done with the CC also. So start from the centroid and go through the entire 360 degree to understand how the shape is varying. Okay, so that is what is that is what is plotted here. You can see how the shape is varying. It is having some kind of how the distance between the centroid and the regions are. So this is the variation of the lateral ventricles and variation of the corpus callosum. And for this image, what we do is we are, so this is, you can think about this as a signal, right? So as a shape metric as a signal. So what we did was extract the Fourier coefficients of this shape signature, and we try to analyze them. Now, again, I am giving you some ideas wherein you can use some deep learning techniques. We have tried to use some deep learning techniques on these shape signatures, on these methods also. So on these lateral ventricles and the corpus callosum also. So like this, we can extract shape based metrics and use deep learning models for further analysis. I am going to tell you how we have used the deep learning model for that. Okay, so we first try to understand how the Fourier transform based features are. Now there are many features that have been captured. Like we have used some about 94 features from the Fourier transform, and 27 features from the corpus callosum. Fourier transform features are being shown to be as significant, and in LV you can see about 94 features are being significant. Okay, so completely we had taken about 250 features. Okay, among them 94 and 27 are significant in differentiating these conditions. Now the next thing is we are interested in doing another thing. So for this Fourier transform, I'm going to calculate the fractal dimension value. So for the fractal dimension value, what we are doing is we have a log-log plot. That is how the fractal dimension value is calculated. So you are plotting a log-log plot, and we are fitting a linear line across the plot, and the slope of this linear line is taken to be as a fractal dimension value. Okay, and this fractal dimension value is calculated for all the subjects ranging from normal to AD. And we are seeing how the variation in the mean values of the subjects are. You can see this is the feature representation plot. Okay, and I can see that the mean value is different for different stages. The mean value is different. Okay, so that shows that this Fourier transform shape signature based approach is also a very good technique that can be used to understand the EMCA condition. Now, here only we played a different part. What is it true that, okay, individually we are getting some good variation, but is it true that two structures are actually dependent on each other, saying that whether corpus callosum and the ventricles are having a relation with each other, like whether these two go hand in hand. So we wanted to understand the aspects of ventricular and callosal features, how they are actually interrelated towards each other. So what we try to do is fusion of these ventricular and callosal features. What we generally do is concatenate these two. Okay, so that is a basic step. Concatenate these two and find out the how it is actually related. So we have different techniques for this concatenation: linear concatenation, deep learning based concatenation, and there are different techniques. So in deep learning itself, we use different kernels for capturing the intricate dependency. Okay, so right now whatever we have done here is two. One is called to be as CCA, canonical correlation analysis, and the second one is deep CCA, or deep learning based canonical correlation analysis. So what we do here is we give the features that are being extracted to a deep learning model. Okay, it's a basic deep learning model with ANN network. ANN network means I have a input layer, I have several hidden layers, and I have an output layer, right? So what we do here is we take all the features, we give it to a deep learning model. The deep learning model will extract the details of those features which are being given as an input, right? So the extracted details, which are called to be as deep features. Okay, so deep features means the deep learning model extracted features. They are actually concatenated, they are analyzed, and they are seen to, they are seen to understand how these features are actually related to each other. So that is what we have tried. Now, there are different significant features that are being extracted from LV and CC, which are being fused using CCA and deep CCA based approach. And we had seen that these features are actually performing very well when we compared with individual features of ventricles and corpus callosum. These fusion techniques are actually very good. They can extract a good amount of information, discriminative information between the different subject groups, that is healthy EMCA, healthy versus LMCA, healthy versus AD. So healthy controls can be seen to highly differentiated from the EMCA subjects. This can act as a very good precursor. Like we can call these this kind of pipeline as a precursor to identify whether that particular healthy subject is actually prone to a EMCA condition or not. Okay, so that is a main thing. And these deep features, we have extracted deep features, right? So these deep features have been given to a classifier, and we have taken two classifier here. I'm just giving you a representation here. We can take any classifier, and how we can actually use ensemble classifiers and other things. We have taken here a ELM based classifier. ELM is an extreme learning machine classifier. Actually, the advantage of this classification technique is it will actually prevent the overfitting. Okay, so we know that very well in deep features, whatever we are taking, overfitting is a very a significant problem, right? So overfitting is nothing but my data. If I have a nonlinear data, okay, nonlinear representation of the data, my model is trying to fit exactly towards the data. It is actually memorizing. I can call overfitting to be as memorizing. It is memorizing whatever information that you that is given you. There is a difference between memorizing and learning, correct? So memorizing means you just I say rata market, like we are taking in whatever notes we have. That's it. But learning is different. Understanding the concept, understanding the technique, that is what is learning, right? So any classifier has this problem of overfitting. And ELM classifier is actually specifically developed to prevent this overfitting by using certain techniques. Okay, so what we try to do is we use these ELM classifier and we try to understand how it is actually performing on the deep learning based features and deep learning based fused features. Fused features means I am taking the two ventricles and corpus callosum, I'm fusing the significant features in that, I'm putting it to the deep learning model, and I am using the deep learning features to a classifier. Okay, so somebody had asked me how ensemble techniques work, like this only. So you taken multiple things together and you put it together and you finally get the outcome. So in that we have identified that the performance percentage or accuracy is about almost about 80%. So about 80% we are getting for classifying between HC and EMCA. So in all the three cases, and this is another variant of ELM classifier. This is also performing somewhat similar, and it is showing a very good difference between the different disease conditions. So this particular pipeline is very, very novel and very can be integrated with different kinds of MR machines that can be used for predicting whether the particular person is having a possibility to have to getting developed an Alzheimer's condition. Okay. Now, the next thing is about different concepts. So I will just take some time. So it's 3 o'clock. So I will take some 10 to 15 minutes to explain how we have, this is completely a different thing, not neurodegeneration and other things. So I just want to give an introduction to those who are working in microscopic images, how we can use microscopic images to enhance the further image processing, or how to use it for disease diagnosis and other things. So what we had done here is I had taken two things. One is fluorescence image and the bright field images. So we are looking at the segmentation approach. Okay, so what is this segmentation approach? Now, so this is, I am not going into depth with the image acquisition part. Okay, rather what I would say is quickly, this is a plate which is actually used for different kinds of different kinds of cellular techniques, like you want to do a gene profiling, you want to do drug discovery, drug testing, so and so forth. We generally use plate-based technique, well plates, wherein we have different wells. The solution is being poured, that is the cell solution is being poured, and it is being mixed with different drugs, different dyes and other things. And we want to image the whatever is being happening, whatever is the reaction that is happening with the cellular structures. So now this particular database is being developed for identifying cancer cells. Okay, how the drugs are reacting with cancer cells. And we have different kinds of cellular structures that are present within the within this particular that are present within the particular well. Okay, so what are the different cellular structures? We have nucleus, we have endoplasmic reticulum, we have nucleoli, RNA, actin, proteins, and mitochondria. All those things are there. And so we are considering mainly nucleus and cytoplasm. That is, we are mainly interested in how the nucleus ratio is changing as the cancer is progressing. Okay, between the normal cells and the cancer cells. The second one is I am also interested in how it is related to bright field images. So what we did here is the images between there are two types of images. One is bright field images, as I told you. The second one is fluorescence images. The bright field images are taken here. This is the bright field image. You can see here nothing is visible, right? So not much difference has been seen between nucleus, nucleoli, and other things. Whereas if you can see here, the nucleus is represented separately, endoplasmic reticulum is there, nucleoli is represented separately. So all these things are available. So this is called as fluorescence image, wherein you are tagging the cells with a particular fluorescent dye. Okay, and then that dye will take up the will be absorbed by the particular structure, and it will be emitting the fluorescence. Okay, so that is why it is called to be as a fluorescence imaging on fluorescence images. And bright field images are not dyed or anything. We are using a simple microscope, optical microscope, to capture these bright field images. So we are using 3 planes images. Like for example, I have a particular what I'm taking is there is a particular focal point that we call in every. So for example, if you're taking an image, a phone also, if you're taking an image, you click on a particular area where you want to focus on, right? So that is called to be a focal point of that image, of that sensor. Like your camera sensor is there. So a particular distance between the camera and sensor and the object is called to be as a focal distance, correct? So here also we have focal plane in a microscope. So a particular plane will be in focus if you set it in that particular setting. So what we are trying to do is, you think cells in the well, they are placed on the slide and they are 3D structures. Cells are 3D structures, right? So when we take the cellular structures, there are different planes of focus that are available. So what we try to do is we try to vary the plane of focus. Like the first plane of focus is on is exactly at the focal plane. The second plane of focus is 4 mm above the focal plane, and the third plane of focus is 4 mm below the focal plane. Okay, so 3 images we are considering. Now, if you can see the images now, when we are taking, you can see these images, they are not very highly visible, right? Illumination is not very good here. So mainly we go about pre-processing in these images. We have some kind of illumination correction. So there are illumination correction functions that are available. So we want to do a correction for illumination, and also we do the focal plane selection. So we want to understand which focal plane is good enough to capture the changes in the images, okay, in the nuclei and in the cytoplasm. So, and the next thing is, after selecting the focal plane, we go about segmentation. So in this particular example, I have shown you a spin transformer. Okay, a spin transformer is a modified unit block. Okay, in unit we have an encoder decoder. It's an encoder. Both are encoder decoder architectures, but in unit we have the encoder to be as a basic CNN model. Okay, here instead of a CNN model, we have a spin transformer block, which is a modified block based on the transformer architectures. Okay, so we have a encoder with a spin transformer block that is being repeated, and we have a decoder. Okay, so what is happening here is, if I give a bright field image as an input, I want to segment the nuclei regions in the bright field image. Okay, so now you imagine I am going to give this as the input, these 3 things as the input, and I want to get these images. Okay, now fluorescence images, I told you we are actually doing a dye-based process. Okay, so it is very complicated and it is costly also. So is it possible that from the bright field itself, we can get the information about the nuclei, cytoplasm, whatever it is, using the segmentation technique? Okay, so that is what we are trying to do here. So these are the representative fluorescence images. So you can see for different genes. So AURK gene is here and RAF gene. You don't have to worry about these things. These are different gene-based cancers. Okay, so genes that are being involved in the cancer-forming cells. Okay, so now how the nuclei of is varying in different stages, and how the cytoplasm is actually varying. Okay, so that is what is represented here. You can see that there are the compounds, that is these kind of compounds are having an effect, that is membrane bulbing is there. There are differences in the intensity values, distribution of the intensity values. There are reduction in the cell volume. So different kinds of effects are there on based on different different embeddings and different compounds that are being treated with the drugs, right? So now what I am interested in is segmentation. That's what we had discussed. So we have different focal planes. There are focal plane 1, 2, and 3. I told you about this focal planes also. So each focal plane will capture different regions of the interest. Okay, so here we can see that the focal plane 1 is having very poor contrast. Okay, and nothing is actually visible. You can see there are actually cells here. You can it's very limited visibility is there based on the cellular regions and the background. And what we want to do is we want to select which among this focal plane is good enough. So first thing is we are doing a illumination correction. You can see how the illumination correction works. So this is a raw image and this is the illumination correction image. This is in fluorescence aspect and this is in bright field aspect. Okay, so raw image and the illumination correction image is given here. Now illumination correction has actually enhanced some of the regions, but still it is not good enough. Okay, so normal image processing technique will not work in actually segmenting the nucleus, cytoplasm, and other things. So we want to go in for a deep learning based technique. So for going in for deep learning based segmentation, we want to select what focal plane is good enough. So what we try to do is we try to understand the sharpness information, and we want to understand how the density of the cells are in the focal plane, in that particular focal plane. And we can see that the focal plane 1 is actually having very good very good sharpness value. Sharpness, if it is high, means it is having good information, right? And also density is also higher for focal plane 1. So we are taking focal plane 1 as a input for the further process. And we are using, as I told you, we are using this particular architecture. So this the focal plane 1 is given as an input, and it cannot be directly taken as an input to the complete block. So what we do is we give it in patches. Okay, why is this so? Basically, whenever we take any microscopic images, it will be of huge size. Whatever images we have taken is of like around 1024 cross 1024. You can imagine the size of it. So 1024 cross 1024, if we are using that as a input, then it will create so much of problems in the process of segmentation, right? So what we generally do is we patch it. We divide it into multiple windows. We take a small window, we give it as an input, we compute the output, and then like this, we do it for all the patches and then stitch it together. So you can get the complete image. So like this, we are doing the images. You can see a small patches represented here. This is all for nuclei. So for nuclei, this is a segmentation in fluorescence image, but I am actually interested in bright field images also. So we will see. So segmentation of nuclei and cytoplasm in the fluorescence image, how it is. So it is actually segmented very nicely. And how the segmentation performance is, we have compared different architectures. So customized CNN, UNet++, Deep Lab, Swin unit. So all the architectures we are actually compiling, and we are trying to understand how it is performing. So the performance of the Swin unit was actually better than the other architectures. So that is why we had taken it for the further things. And how it is actually varying, how the segmentation performance is, we are actually getting about 87% accuracy in matching the areas between bright field and fluorescence images. And this is also a loss function. You know that loss function and accuracy are an important metric to evaluate any model. Okay, so that is what is represented here. And the correlation matrix is 87% we are getting, and the accuracy with which it is segmenting is about 98%. Okay, so this is what how the metrics are related. Now, so the same is actually done for bright field images also. So you can see here, bright field images are given as an input, and the segmentation is being carried out. So you can see the segmented regions and the ground truth. They are being compared. So this is for nuclei and this is for cytoplasm. So the same Swin unit transformer is actually used for segmenting and comparison with the ground truth. So we are getting a pretty good results with bright field segmentation. Okay, so and after that, we also evaluated them using different feature extraction techniques. So I'm not going into detail with this. So different morphometric features, texture features are being extracted, and they are being compared for the two classes, that is AURK and RAF, I had told you, right? So they are being compared for two classes like this. For cytoplasm also we have done. And we see that the differentiation accuracy is pretty much high when we are comparing the texture-based metrics rather than morphometric metrics. And it is actually fluorescence images are good enough for that, but it is still bright field images are also giving out good sensitivity results. So they can be used as a pipeline to further understand the problem with classification and other things with respect to the disease condition like AORK and RAF, like that. Many different drugs are there, so that we can test, we can use them for segmenting and classification. Okay, so this is so I have just given you a very brief overview of two different topics that we generally work on in our lab. But it is not the only thing that we are working on. Unfortunately, I don't have time to explain you what are the other things that we are looking at. So there are different projects that are happening in our lab. So I have given you an overview. We do work on image processing, brain tissue parcellations, and there are different techniques that we are working on. And in pattern recognition, deep learning, machine learning, model development, device development, medical device development, electrode technology, and we also work on some image-guided intervention, like how navigation can be done using vision-based navigation, how they can be used to steer the endoscopic endoscopic cameras within the body. So all these things we are actually focusing on, and our lab basically has a motivation to translate whatever research that we are doing into a commercialized product. So we are also working towards that. And I would request, if anybody, any of your students are interested in joining our lab as a PhD for their doctoral studies, or they want to, we also have a opening for JRF. So if they are interested in joining JRF, also they are quite welcome. And please ask them to. So the link is given here. For I will also paste the link to the chat window. Okay, please ask them to apply. And if they are interested, please ask them to join the lab. They will be, I'm sure they will be learning many things, and they will be getting an overall exposure in the area of computer vision, machine learning, deep learning, and image processing. And it will be very good. And we also have an opportunity to convert the particular JRF position to PhD, so they can actually do their PhD along with their JRF here. So if anybody's student is interested, if anybody is interested in doing a doctorate, please mail me. You can contact me through my mail ID. My mail ID is given here, rohinip@IITDM@ac.in. Okay, so please do contact. And if you have any doubts, you can ask me. I can answer as much as I can. Yeah.
Yeah, thank you, ma'am.
You.
Yes, is there any question from participant side?
Hello.
Yes, Miss Jaya.
Am I? How to get the medical images, madam? Where is the data set available?
Okay, there are many data sets that are openly available. See, the problem with medical images are it requires ethical clearance. So when we want to acquire any medical image, we need to have a support with the doctors, or we need to have any collaboration with the hospitals. But
Okay.
to start with, like if you want to start a research, or if you have any hypothesis, like this is actually important, and it will have a good research aspect or good productization aspect in the near future, then what we can do is you start with the basic images that are available online. Okay, so there are many different data sets that are being publicly created, and it's being open to the research community. So we have, whatever I have shown you, the images which they are available online. So you can check out the resources, and you can see whether whatever hypothesis you are considering, whether it will fit into your, whether it will fit with the images that are available. Sorry? Sorry, I did not hear your question.
Can you provide the links where we can get the images? Links, online
okay,
link.
you can search for ADNI, ADNI. ADNI is a database for that is widely available for Alzheimer's disease.
And in the chat box, no
similarly,
spelling. I'm not.
ADNI, ADNI, Alzheimer Disease Neuroimaging Initiative. ADNI. I can write it for you in the chat. ADNI. This is the.
Your voice is breaking, madam. Hello.
Am I not audible?
ADN. Yes.
ADNI.
ADNI. Okay.
Yeah. Okay. This is a short form for Alzheimer Disease Neuroimaging Initiative. So you can check out the website for getting images. So they are available, free, openly available. You require to submit a proposal wherein you need to give what is the research that you are going to do with the images that you are going to take. So if you can give an overview of that, then they will provide you with the images. Yeah.
Okay. Thank you.
Yeah, is there any more question from participant side?
Thank you so much, ma'am, for this enlightening and insightful session.
Thank you.
Any more questions from participants? Your expertise and in-depth knowledge truly enriched our understanding, and we are deeply grateful for the time and effort you have taken for both the sessions which we had with you yesterday and today. Thank you, ma'am, once again. Nitin sir, shall we move ahead?
Yes, ma'am. Thank you, ma'am. Thank you, Rohini, ma'am.
Yes, sir. Thank you very much, sir. Thank you.
Yeah, for joining and provide informative lecture on medical imaging.
Thank you very much.
Yeah, and neuro.
Thank you for the
degenerative
opportunity.
disorders.
It was great time, and I thank Dr. Nitin Sharma sir. Thank you for giving me this particular opportunity, and hope to meet you, sir. Yeah.
Yeah.
Thank you.
Now, as we move ahead in the session, it is my pleasure to introduce our next speaker to you, Mr. Ayush Singh. He is application engineer at Integrated Microsystem, where he specializes in providing technical support for Synopsys Simpleware software. He works closely with leading scientists, doctors, and professors from prestigious institutions like IITs, AIMS, NITs, and government laboratories. His focus is on helping researchers unlock the full potential of Simpleware's advanced capabilities for 3D image processing, model generation, and analysis. This support plays a key role in applications such as medical device development, diagnostic imaging, and personalized treatment planning. Today, he is going to discuss with us industry-based applications of medical image processing. I welcome you, sir.
I think some technical problem is from user side, so he will join soon.
Dear participants, we are soon expecting Ayush sir to join us. He is facing some technical issues and we will update you accordingly.
During this time, I have a short documentary on Maharaja Agreshan Institute of Technology.
I'm audible?
Yes, yes, sir.
राजश्री सोल से सारे अपडेट्स पाने के लिए बेल आइकन दबाइए।
भगवान श्री कृष्ण के समकालीन महाराजा अग्रसेन
Yes, Ayush, sir, you can start your session.
Yes, sir. Sure, sure. Let me share my screen, sir. I think my screen is visible.
Yes, sir, it's visible.
So, the presentation here. So, a very good afternoon to everyone, to every participant, to the organizers of the particular FDP, organized at MIT. So, this side Ayush, I am an application engineer at Integrated Microsystem. Today, I'm here to have a particular demonstration session of the advancement that are going in the 3D image processing using Synopsys SimpleWare software. Earlier on, in the previous section, the inaugural session, the CEO of our company, Mr. Praveen Sikka, briefed us about the Synopsys SimpleWare tool and the particular image processing, 3D image processing sector, which has been growing tremendously with the passage of time. Since the particular growth rate is much more faster, so what we have is we require a solution that can pace up with the particular growth rate that we require for. So Synopsys SimpleWare software bridges up the gap between these two. And here in the particular section, we shall be delving on how usage of various automated algorithms does help us in reducing the time of computation and why the particular less time that we have in the competition thus increases our accuracy as far as also decreases the time for any surgical planning. So without ado, let us start with the session. So let me introduce with what is Synopsys Simpleware and how it can be useful for our work. So basically, Synopsys Simpleware is a 3D image processing tool by which we can generate model. We can do machine segmentation, etc. of data obtained from CT scans, micro CT scan, MRI, etc. Apart from these standard medical imaging dataset that we have, we also have support of additional raw image dataset, JPG, PNG, TIFF, and many others. Why is image formats are of importance for us? Because if we have a proper spacing and we have a proper stack of 2D image slices, we can convert that out into 3D model. Once the particular 3D model has been converted, we can have it in various sets of export formats. So we can generate finite element model, we can have computational fluid dynamics model generation, NURBS model generation. Apart from it, for 3D printing and all, we also have STL 3MF support. So here are the particular workflow that we use in Simpleware. The workflow in Simpleware is quite simple. Here, what we have is we call it a 4-step workflow. What are the 4 steps that can be of a concern? So the first one step being the image processing. So in the image processing, there are primarily three sectors. First, we need to define the region of interest that we need to work on. Apart from that region of interest defining, we can also do the cropping operations. We can do the resampling, rescaling operation to upscale our data set if there are so many distortions and the data set is not proper. Apart from it, we are also provided with various advanced filters to correct our CT data, MRI data that we have provided in the software. Also, if we have any alignment change in the image data that we have provided, that also can be corrected within our tool. Once our pre-image processing sector is done, and we have the processing step. The image processing step comprises of the segmentation. By segmentation. So in simple way, we have three types of segmentation. The first one being fully manual segmentation, the second one being semi-automated segmentation, and the third one being fully automated segmentation. How we use this particular segmentation is quite a good task. Why so? Because whenever we provide the data set, it is not that if we have taken a pelvis CT, so we would be only having that. Apart from that pelvis CT, we would be having nearby noises, nearby surface capture of various skin, muscles that come in between our particular bone data set that we need to define for. So to work on the specific bone part, what technique that we use are watershed algorithms. Then we check on the grayscale value, that is the Hounsfield unit. So every element in any structure material is comprising of different density. To segment out it on the basis of density, we use the particular grayscale value segmentation. And corresponding to that, in the semi-automated segmentation, we select a particular grayscale range. That, for example, if our data has a grayscale value range from 0 to 255, and if our bone part lies between 120 to 255. So we will specify that particular specific region being 120 to 255, that between this region we have our bone. So please segment the particular file for us for the specific part. Once this basic segmentation is done, we further split down. So for example, if since we are taking the pelvis CT example, so herein we have 5 different parts in the case of bone structure that we can segment out. The parts namely being left hip, left femur, right femur, and sacrum. So we further need to do the segmentation to the individual parts. As if our requirement is only with the right hip, then all the rest parts that we have created from segmentation might not be of any use for us and will only be occupying the data set memory, thus reducing our computation capabilities. So to specify that if we have a particular acetabular cab implant for a right hip, that can directly be done by isolating the particular part out. Once this is done, and we also have automated segmentation algorithms. So for automated segmentation algorithm, it is the pre-trained data sets that we have worked the software on, and we are still on the particular development phase, although it is in the market. In the automated segmentation, we have two aspects that we have covered. That is the automated segmentation for orthopedics, as well as automated segmentation for the cardiovascular data set. In the case of orthopedics, currently we have knee CT, ankle CT, CMM, that is craniomaxillofacial CT, and then we also have particular knee CT, ankle CT, and various other tasks that we can directly segment out in the case of orthopedics. Apart from it, in the case of cardiovascular, we can do heart CT segmentation, and once the segmentation is done, we can also do the heart valve analysis. These are all very important for us because we need to reduce down the surgery time to increase the particular efficiency that is being required from us as an engineering point of view. Apart from it, we also require various measurement tools to do proper measurements, thus we can get a precise result. For example, every anatomy is different when we are dealing with a particular medical image processing. So to work on each and every individual's anatomy, we need to have their CT data. Then if a particular person has a particular implant design for knee, so the particular same implant design might not work for any other person as the anatomy is different. So we need to have customization option. In that case, we require the patient-specific implant design. To do so, we use the simple way and its capabilities to import the surface to develop the particular implant to connect with the particular bone surface and manual. So what we can do is we can do a virtual surgical planning of the same. In what cases simpleware is used for? So we have a medical image processing. So we have a large type of import and export support. Apart from it, we have a full integration, right from manual segmentation to fully automated segmentation. So what we can do is we can do fully automated segmentation, and later on, if we require to do further manual interpretation, that is also possible in simpleware. Apart from it, we also have the vice versa, that is being a possibility. So we can do manual segmentation and then we can run the automated segmentation for the specific part that is required. The second one being the SPL, CMF, and CAT file support. So we do have large number of export formats supported. Apart from it, we have interoperability with other software such as ANSYS, Abacus, Console, MATLAB, VTK. So whatever resulting output that has been produced in our software, we can export it out for the particular files. Why this particular FECFD model may be useful for us is we need not require remeshing in the particular software, and we can directly run the file and thus produce the output that can be again visualized in our tool. That. Apart from it, we also have fully customization option. So we have inclusion of scripting and GUI. So whatever task that we do, that's recorded in a form of script. We can do part scripting. So if we only require, for example, smoothing of a particular part, and we had created an automated script for that, and we need to run on hundreds of data sets. So we can do it all at once using the Python scripting option or C# scripting option. Apart from it, we can also have our own APIs creation for various sets of works that we require for. There are some application areas that we use simpleware for. The first one being the implants, guides, and surgical planning. So we have predefined tools that help us in doing the particular implants, guides, and surgical planning. We also have cutting guide tool to directly create cutting guide surfaces over the existing surface, which can be of very beneficial in the case of any orthopedic part. For example, if we need to have a knee specific implant design, so we can take the reference from that and we can directly develop the surface accordingly. Apart from it, whatever results that we produce are highly accurate results and compatibility is with ANSYS Abacus console. So we can do the particular analysis and we can rely on the particular simulations that we have conducted for. Thus, the accuracy that we have is quite high. Apart from it, we also have point of care 3D printing. So we can have individual parts to be printed and a real prototype of the actual anatomy can be produced in front of us. There are some more application areas in the case of life sciences and clinical application. So in the case of orthopedics, we have automated and semi-automated tools to segment out anatomies. We can combine CAD implants and anatomical data. Also, we have an option of accurately quantifying the bone geometries and export the particular multi-part FECFD meshes to various solvers for doing the simulation. In the case of product design and analysis, we can do customize the particular product design to individual anatomies. Then we can virtually check fit of designs on the human body, export models to simulate the various real-world performances. By all this means, we can get a better insight for future products by analyzing the real sets of anatomies. When coming to the physiological flows, we do have its option enabled for both cardiovascular as well as respiratory systems. We do have an option of reslicing images along vessels and airways. We can combine anatomical data with stent models, set mesh boundary layer and custom inlet and outlet for fluid flow analysis. And we can also quantify vessels with the center line network tool that is one of our headlines in the measurement section. Then, in the case of EM and neuromodulation, we can integrate CAD designs like MRI coils or electrodes with the image data. We can also use very easy to use registration tools for positioning of devices and generate and export various simulation ready meshes. Apart from it, we also have its advantage in the medical device research and development, wherein we can rapidly integrate CAD devices into CT MRI scans. We can research for human device interaction. We can have automatable, repeatable operations with the help of scripting. And thus, we can have it in various medical devices creation. Apart from that, we also have it support in the 3D printing preparation. So the files that are produced using simpleware surface tools are high quality watertight STL files that are directly ready for 3D printing and does have support of various export formats such as 3MF, STL for 3D printing. Then we also have support of virtual reality, WRL, the VRML formats. We can also add internal lattice structure to reduce the weight of any surface that you're working on. Also, we can use this particular models for various teaching aid. So these are some of the features of SimpleWare as discussed earlier. We have support of both 3D image data as well as 4D image data. So whenever we are doing an segmentation using the automated segmentation or manual segmentation, in the case of automated segmentation, it would already do a particular segmentation for each and every slice view. In the case of the time offset as well. So we can have the particular mimicking of the particular beating of heart directly within the software. Apart from it, we have great volume rendering tools to check for the particular volume rendering, and then the particular results that are produced are very high quality results. Apart from it, we have various surface tools option. So we also have interoperability with SolidWorks. So whatever designs of implants that we have created in SolidWorks can be live visualized in SimpleWare. Whatever modification, dimension change that has been required can be positioned out. Apart from it, we can also create knob surface that is non-uniform rational B-spline structure model, and then export it out in a step or IS file that is required for a specific workflow. In the case of automated segmentation, we also have key landmark points which we use for easier measurement. It automatically identifies the particular key landmark points. We can also move or change as per if we are not satisfied with the particular landmarking points that it has produced. Then we also have various volume matching tools, so we can directly create high quality finite element models or computational fluid dynamics model. We can set for the contact surfaces, material properties, whatever is desired by us. We can also set in the particular boundary conditions type, then node sets, cells, match refinement of any region. These are all the particular possibilities that we can further cater for. In the case of measurements, we do have point-to-point measurement, we have angle measurements, then we have a wall thickness tool for a whole model that we are going to produce. It will create a particular wall thickness range. So it will tell us minimum and maximum annotation point, as well as the particular range that we are having of the wall thickness all around the particular model. Then we also have various custom templates that we can further use for. So we can create our own set of specific templates, and then we can work on the specific parts. For example, if we require a center line analysis, a core analysis, that can be directly achieved in SimpleWare. We also have various shape-based measurements. So if we need to measure the distance between right femoral head and left femoral head, we can directly insert two spear shapes and do the measurement accordingly. This all what I am discussing, I will also demonstrate in SimpleWare and how to do so. In the case of 3D printing, we are given with a very enhanced toolkit. Herein we can prepare the particular 3D models that have to be printed out. We can create cut, hollow models. We can also emboss text on the models. Then to check the integrity of the model, we can also apply a grayscale visualization. And if any sort of defect are being present on the data set, we can directly solve out using the inspection model, which works using automated algorithm to directly fix any error that has been present. Then we can export it out to any file that we require. Here is the overview of the latest version, the new features that have been introduced. So we have cutting right tool, we have AI-enabled bone thinning to solidify any bone data set. Then to check for the accuracy of automated segmentation with the manual segmentation, we can also use the mass similarity calculator. We also have hip revision city, so it automatically does the particular auto segmentation for a hip part that also has implant within it, so that we can work on a specific part. Also, it would segment out the implant and position it for us. Then similarly, we have inclusion of spine CT to automatically segment out a particular vertebra, particular spine into vertebra and sacrum. And we also have certain improvements in AI algorithms. Now it captures more surface area rather than the less surface area that was being captured in the previous versions. Here is a particular overview of the cutting guide tool that have been introduced and how to use it. So surgeons can create their own cutting guides on the specific patient-specific models, and thus can create a 3D printing output of the same. So like this, for example, if I take a pelvis data set, I just need to set out the particular track. So I will select the particular plane along which I need to create this track. Then I will set the particular pins and drill holes. Herein we can set the particular radius, height, then ball width of the particular pins and drill holes. and thus we can place it. It would always be perpendicular to the surface in any scenario. Once it is set, we can create a base and it would create us outer surface on the existing model that orthopedic model that we are working on. Similarly, it can also be used in the case of cardiovascular for stent model designing and all. Herein, we also have AI-enabled bone filling. So when we do the manual segmentation and semi-automated segmentation, we might not get very accurate and highly optimized result. So to cater that, what we have is we can just have a single click operation of apply, and it would automatically solidify the bone. The the results can be seen over here. So this is the manual segmentation before bone filling has been used, and after the particular bone filling has been done, we can see the enhanced results that we further obtain. That can also be checked in the 2D view by seeing the quarter levels of the same. Then we also have this mass similarity calculator. So it works on dice score calculation to calculate the accuracy of AI-enabled segmentation versus the manual segmentation that we usually work on. Herein, we can also check for our set of accuracy, how we are doing that. And to further check it, we can also enable background volume rendering to check for the same. Then we have this prevision CT. Now it reduces any sort of metal artifacts that have been present. Also, if, for example, in this case, the proximal femur implant has been positioned. So it will also segment out this particular hip and segment this particular proximal femur out. Also, the plus sign that we are seeing over here are the particular landmark points that would be automatically captured. Then we have the single inclusion for a spine dataset. We just select the CT data, click on a single click operation, apply, and it will automatically do the segmentation task for us. Herein, we also have one more option of this keep all bone fragments. How this particular keep all bone fragments is useful for us is if we are given the trauma dataset or a fracture dataset. It would also keep the nearby grayscale variation in mind and do the segmentation. And thus, it would not clear out the particular fragment of bone that are being present in the trauma dataset. Herein, we also have certain improvement in the old algorithm that we used to have. Now it captures more surface area, and it's now much more accurate before the particular attack. So this is the particular system requirements. So it runs on both Windows system as well as Linux system. Apart from it, we also have its system requirement of processor of 7th generation or higher, a minimum 16 GB of RAM is required. Then a normal graphic card around 4 GB is quite sufficient for us. So does anyone have any questions, or I can start with the live demonstration?
Yeah. Any question from participant side?
So let me start with the live demonstration. Meanwhile, if anyone has any queries in between, they can also ask them. So this is the particular GUI of the tool. Here in this is the latest version that we are currently using. So we have version 24.12 that is released on 11th of December. Herein are all the import formats that we can import the data from. So we have DICOM dataset, so MRI, CT, micro-CT. All folders can be selected from here. Apart from it, if we are working on any industrial datasets, we can also import it in the form of DICOM. Then we also have connectivity with a PAX server, so we can directly enable it and take the data from any hospital server if we require for. Apart from it, we also have a stack and raw images support. So we can click over here, and any raw image dataset that we are having in a format of a BMP, JPG, PNG, TIFF, we can also import the particular files in the exclusive formats. Then we also have 3D image format support, so .str, .hpm, MSG, then IMG. So all are the 3D image format, 3D images that we can directly import for. Apart from it, we also have support of STL and CAD files, so we can also load the particular STL and CAD files, do the modifications that has been required to do any sort of surface editing, embossing of the test, changing the internal lattice structure. Once this all is completed, we can again export it out in our specific requirements. Apart from it, if we need to import any volume mesh file that we have computed in another software, that can also be done. So we will just select the particular database from which the file has been taken, and we can do the particular import. Herein is the project information section. So whenever we will import a particular image data, we will be getting the particular spacing of the particular images, the image extent value, the total extent, the object counts that it is having, the memory size that it is requiring. Here is the open project section, wherein whatever projects that we have created, we can load it from here by going to open projects. Apart from it, we also have a dedicated help and support. So here are all the particular keyboard shortcuts that we can use while we are working with any data set to reduce the time that we require for. Apart from it, we also have a dedicated tutorial guide. So in this tutorial guide, it is mentioned in a very efficient way how to use each and every tool, in what scenarios, which tools can use what is the algorithm behind any tool. So to understand that, we have a detailed explanation using pictorial representation as well as GIF representation to how to do the particular modeling, how to create an interface, then to do any sort of model creation. We can all go in this particular tutorial guide and we can check out the same. It's a very detailed tutorial guide, to be honest. Then we also have a simple web website wherein we can check for the publications, the results, the papers that have been published, the new features that are being introduced in the latest release. Like we have introduction of Monai model, total segmental model in the latest release, so we can have external models used for automatic segmentation. To showcase any data set, let me take a particular spine CT data set that I worked on recently. So this was provided to us by one of our customers. Herein we had worked on the particular specific data set of our deformed spine. So this is the particular data set option that we get. Herein the patient information which has been anonymized. Herein we can get the particular images, the total number of images in this data set. That is, the total number of slices are around 533. The modality of this data is a CT, and all the particular information has been anonymized over here. Once it is done, we will just click on finish. And once we click on finish, it would load us our data set in 4 views. The 4 views that we get are majorly 3 2D views and one a 3D view. So this is just a 3D view. This is just a volume rendering of the data set. It is not yet a model, not yet a mask. It is just a volume rendering of the data set. We can play around with this particular volume rendering. We can go over here. We can change for the presets. Then we can also define our own setup point. For example, if we just need to focus on bone, we will select the bone preset and it would just highlight the particular bone region in the background volume rendering. So this is the particular volume rendering. Apart from it, if we are given with a soft tissue data set, so we can see the particular blood flow by using this MIP rainbow. So to highlight the specific region, since this is a particular CD data of highlighted bone, gated bone, so we will be just getting a pictorial representation. But if we have any arterial flow data set with us, we can also check for the particular flow directly within the tool. Apart from it, whenever we get the particular 3D, just let me load the particular preset as well. Yeah. So whenever we are working in this particular view, we go over to this image processing section. In this image processing section, we have the option of resampling. So we can resample the particular data set, whatever is required by us. So we can go over here. We can do the upsampling. We can do the downsampling. We can see that right now the data set memory size is around 280 MB. If I tend to upscale it, so let me take a cubic and isotropic value. Once done, so we will be seeing the dataset memory size increases. Also, currently the slices are 533, and so it has started from 0 to 532. So it has total number of slices of 533. Once we will click on apply, it will do the upsampling of the dataset. It would increase the memory size. Thus, if we have a clear dataset, so the upsampling option is not very much recommended. Here, it will take us around 1 to 2 minutes to do the operation. Meanwhile, let me try to connect the faster system. So this one's a quite fast system. So let me open the same tool over here.
Sir, how you choose the parameters of the software?
Like what's it? What sort
Different
of parameter?
parameters you are taking for analysis purpose.
Like the city data that has been provided, sir.
And sampling rate and these things you are giving, or automatically it will take by
It
the
will
software?
it will automatically detect the particular sampling rate, and also we can modify the particular existing values that we are having, but it would automatically detect the particular samples for us. So let me take a different example to showcase much better. So
And how much accuracy of the detection of the disease of the software?
so it's quite accurate. We have certain papers that have been published in which it has been showcased the accuracy is around 97 to 98%.
A real-time application is there,
Yeah, real-time applications use them.
and you are achieving this 98% of accuracy.
98% accuracy using the automated segmentation one. So whenever we load any data set, sir, either we go with the manual segmentation. So wherein we use all the transformation options, the segmentation, the morphological operations, the additional such as smoothing filters. But this all have been incorporated in this automated segmentation one. Herein we just select the particular hip city data, and whenever we will click on this keep all bone fragments, even if our data has a fractured data set, it will identify the particular data set for us. We can check on the time. So right now it's around 3:53. It would take around 2 minutes to segment out this particular hip into left hip, left sacrum, right sacrum, proximal femur. So all the particular parts can be identified. It would take just around 2 minutes, and once it has been segmented out, we move on to the next part. Apart, if we don't require the manual segmentation, we also have various sets of semi-automated operations that we can further use. So for the same data set,
With the help of this software, you have provide some early detection of the
yeah,
disease.
early detection of the disease can be done, sir. So if we have any CT data set with us, we can definitely work on it. And for example, any tumor detection. So we can, since it will have a very different VSK value, so that can be achieved. Some of the customers are working on that field using this tool. So it's running in the background. Meanwhile, let me
How
load the
much
same data.
how much early detection you are achieving till this time?
So we don't have exact number with us, but we have it. So we have one of the papers that have been published in the international market. So it was published by John Hopkins University. Over there, they had done an early detection of around 7 months, I think so. So they had detected 7 months earlier the particular tumor.
For which disease, sir?
So it was a brain tumor detection, sir.
Okay. And for cancer detection of
Cancer
different type of
detection
cancer
can,
detection?
sir, it can be done. It just depends on the particular end user of the product. So the software caters many parts. So we have 3D printing of any anatomical data set. We have the particular implant positioning and integration with the data set. Apart from it, we have early disease detection, the cancer models and all. So if a particular customer wants to work on the particular cancer detection, they can also develop their own set of API to automatically detect that within the tool. So automatic extensions are supported in this latest version. So we can create our own set of models, and then we can work on the data set.
And where you are taking the data set?
Pardon?
You are working on some data set, I think.
So this
For
is the particular automated
AI
segmentation.
based.
Yeah. So
You are analyzed on some data set, or only some particular image?
we have analyzed on the example data set that is provided with the software binaries. So we do have certain example data set that we work on. Apart
You are
from
not
it, we have
collecting the data set from in real time.
we don't collect that as of now. The company collects, the Synopsys developer team collects. We don't collect as of now.
Okay. But you are analyzing in real time on the basis
Yeah,
of that data set.
we analyze on the real time on the basis of that data set. So, for example, this is the automated segmentation that has been completed. So now to check the particular accuracy of the automated segmentation, we will just click on this generate a fast preview. Now what we will do is we can overlap it with the particular background volume rendering. Herein we can check, let me change it to a bone. Yeah. Now we can see the particular surface area capturing is quite accurate while overlapping it's a particular volume rendering of the data set. Herein, for the same data set, if I work with a particular manual segmentation, so I just need to go over the same folder. That is the example folder that comes along with the software. So we have it in C drive. In the program files, we have Synopsys folder. And the external models that we support are over here. So for example, if we work on the same data set, we go over here. Yeah, so it has brought down our data set over here. Once it has brought down, if we need to develop our own set of external models that can be created over here and further loaded. The particular external models that we currently support are Bonai models, as well as the MoniAI model, as well as we have the total segmental modeling that is currently supported. So even if we need to create our own set of models, that can be done using the scripting part that we have over there. And how to generate our own set of script is quite accurately mentioned in the particular tutorial guide that we have. Herein, I was showing the example of the particular manual segmentation of the same dataset that we have done the automated segmentation for. We will go to the specific folder. We will select the particular Nikon file. Now, to do the manual segmentation, we generally recommend to use a semi-automated approach rather than fully manual segmentation. Because in a fully manual segmentation, we need to paint each and every region like this on each and every slices, and it is quite a tedious process. We need to work on around 239 slices in coronal view, 278 slices in the axial view, and around 511 slice in the sagittal view. So to do so, what we generally prefer is we prefer a threshold-based approach or a multi-level offset segmentation. And the threshold-based, we just select a particular histogram page along which we need to do the particular segmentation for. Apart from it, in the software we have certain CD presets. So if we use the particular bone, we will be just producing the mask of the particular bone dataset. So to do so, we will just click on here. Let me drag and drop. Yeah, we will click on this fill cavities in Netscape mask. Also, we will here click on the only keep the largest resulting element as we want it as a single model and we don't require any sort of noises that are being trained. So once done, it will be producing as a semi-automated result. Herein, there are many set of further modifications that are being required. So we can compare the result over here. This was the automated segmentation. It has done whole smoothing. It has filled the particular inner gap of the bones. It has also done the bone filling. It has worked on the particular each and individual segmentation. So if we need to just work on it right, we can just isolate it and work over here. And if we need to bring any implant within our particular data set, we will click on this import surface. We will click on implant, clicking on import, and it will bring us in the particular view. Now to bring in our current view, we will just click on the zero position. Now it changes the particular coordinates value to 0, comma 0, comma 0. That is the origin. Once done, we will click on this position and orientation and we can do the further manipulation in the 3D view. So herein we can either manipulate the data set in the 3D view or else we can do the further modifications in the 2D view. To do so, we just need to change the interaction mode from 3D to 2D. And herein we will be getting over here. We can rotate it like this. So this gives us a very nice approach to work with the particular implant to do its positioning with the existing image data without much further work. So we can directly do the positioning over here like this. Once we are done with the particular positioning and all, we just need to do is we need to use a single click operation. So once we are done, we'll just click over here, apply. Once we have clicked on apply, we can move it like this. And it has brought down our particular surface over here. Now to integrate both surfaces in a single surface, what we need to do is first we need to use a dataset conversion. So in the dataset conversion, we do have this mask to surface. In this mask to surface, since I am only working on the right hip part, I will go over here, generate a surface, go over here, generate a surface, and it has brought me a particular hip right surface for us. What's the difference between a surface and a mask? So a mask is the actual representation of the data, and the surface is the particular surface information that we have captured of the particular mask that has been produced. The surface can be integrated, exported out in STL format, CMF format, whichever is required by us. If we need to use certain Boolean operations to connect different surfaces, that all can be done after we had converted the particular existing mask into a surface. Apart from it, if we also require to add internal lattice structure to reduce any weight, what we can do is we can select over here, we can select the coarseness and fineness that we require for null shell correction. Apart from it, we can select the unit cell type, uh, preview cell size. You can also check for the preview how the particular internal lattice structure would look. If we are satisfied, we will just click over here and it has produced us a particular internal structure option. How we will check with the internal structure? We will go over here, we will view it. Herein we can check for the internal lattice that it has created up. So thus, if we are working on any implant or do the 3D printing of the dataset, so it can be directly plotted down and we can reduce the weight also. Apart from it, one more option that we can see is a virtual X-ray resolution capability. So let me go over here in the view section. Herein we have the virtual X-ray. So for whatever CT data that we are having, we can also generate a virtual X-ray over here. Herein we can set a particular title. Also, we can add the orientation levels. We can change the particular orientation also from sagittal, axial. So in different orientations, we can directly generate a virtual X-ray. Also, if we are having an implant, that also we can burn in to have a proper visualization. Once when we can save it. Apart from it, we also have various sets of model creation capabilities. So we can select out the particular mask that we need to work on. We will just drag and drop to the model creation. Here opens us a particular tab of the model creation. Here are all the export types that have been supported for surface model. So we have a 3MF support, we have a STL, Abacus, ANSYS, Point Cloud, MATLAB, Open Inventor. We do have a large support of export capabilities. We also have WRL support for virtual reality modeling language. Also, apart from it, if we need to generate IFE model, we can also do the individual modeling. So if we need to set a particular different coarseness level for proximal femur, for hip left, hip right. So all the particular material property detection can be done individually. Apart from it, since every bone part has different Young's modulus and Poisson's ratio, so we can also define the individual properties of the particular part. We can also save the particular part properties so that we can later on use for the similar data set that we are working on. Once done, we will just click on save material and later on we can load the materials from the library that we have produced. Here in the case of finite element modeling, we have model generation capability of Abacus, Adina, ANSYS, ANSYS Workbench, COMSOL, LS-DYNA, MATLAB, Neston, Veteran, and BTK. Here, similarly, we can generate a CFD model. We can select the boundary conditions over here. We can add all possible, and we can further set the boundary condition type. Apart from it, in the case of nonce model, we can have a step null peg or a I guess num patch file that we have created. So I do have one example with me that I have created in my last meet. So I was working on the particular data set of an ECT data. We had to do automated segmentation for a CAD modification for a total knee replacement surgical planning. And just I will be showing the particular data set after this. So this is the manual segmentation approach that we go. Once we do this, we will go in the split region, we'll select hip, and then we mark out the particular boundaries individually. So if we need to select a hip left, for that we will go over here. So we'll select it hip left, hip right, then sacrum, femur left, then femur right. Once done, we can either preview of the segmented part. If we are satisfied with the pre-segmentation, we can just click on apply. And for example, if I just need to work on the right femur part, so I will again isolate it and further work on the specific region when we are doing the manual segmentation. Once the particular model has been created, we will isolate this particular part out. And now to fill in the particular empty spacing, the cavities and all, we can directly work over there. Also, if we need to fill in certain spaces manually, what we can do is we can go to the specific point. We just need to click on left click followed by P keystroke on the keyboard, and we will be going to the specific point. Now we need to finish it up using the smart paint. So we need to paint it manually, and it would automatically detect the nearby grayscale variation. Once done, we can also check for the result change in the 3D view by generating a fast preview of the same. You can see the surface has been captured. Now we can use various tools such as bone filling tool to solidify the particular bone data set that we are having. Apart from it, we also have various set of smoothing filter to smoothen out the bone part that we are working. Similar to this, we also have the same set of segmentation capabilities for any hard data set. We had also developed a particular prototype model that was used for a particular patient specific and cardiovascular surgery planning to show that data. Just let me move here. Yeah, I do have the particular model. Meanwhile, it has solidified the particular bone data set. We'll just click on this and it will pass for you. You can check over here that it has solidified the particular bone. But still, there are many refinement that we need to do. So we can use the recursive Gaussian. Then we also have a smart mask smoothing operation that we can further do. So you just need to click over here. And it will
one question from the participant side in the chat, that how can we get this software, and kindly share the download file of this software.
So it's a commercial software, but we do have a 30-day trial period. So anyone can apply the particular trial license and then use at their end. I will be sharing the evaluation copy link in the chat box.
Yeah, okay, sir.
Yeah. Apart from it, we also have certain measurement capabilities. To actually, we do have limited time, so I might not be able to cover each and every aspect of the software, as the software has various other tools to further use in the measurement, whatever automated segmentation that we have obtained. So the landmark point that I was discussing of, it would automatically create the particular landmark points. Now to know distance between two different landmark points, we can just click over here, selecting the landmark point. And thus we can get a approximate distance calculation here, and we can move to the exactly on a point, and we can do the overlapping. Once it is done, we can export it all in a form of CSV file or a text file. Uh, Once the particular CSV file is produced, we can go over here. Herein, we can get the particular XYZ coordinate value. We can get the particular grayscale variation corresponding to the specific point. So, like this, the particular reporting can be done. Apart from it, we also have a master statistic template that can be used. So, we can go over here. We can generate statistic of activeness. We can load the template, and like this, we can get the particular voxel count, triangulated volume, surface area, grayscale mean value generation. Then we can also check for the wall thickness. So we just click over here. And since we had selected for only this right hip, let me isolate this out. So to calculate the wall thickness, we will just click on update. Herein we get a particular mapping of the particular wall thickness. Herein we can check for the surface. Apart from it, we can also create a centerline network for the same to have a much more greater measurement using the centerline network tools by going to the specific point. These are all the particular sets of centerline that gets created. Now we can go over to this centerline network tools corresponding to each and every centerline. We can get the curvature, the mean, minimum, maximum mean curvature. Apart from it, we can also have best fit circle for corresponding to any centerline that we are having. Wherein we can also select for any point corresponding to that plane normal value, the plane coordinates, area, perimeter, all can be done. So these all can also be exported out in a form of report. We just need to develop a model. And once our model has been developed, it will generate us a report out for the same, the volume fraction and all. These graphs are empty as I haven't generated a model and done the simulation for the same. Once the particular model generation is done, we will also get the particular graph calculations. So this is just an overview of the tool. I am sharing the evaluation link copy in the meeting link. So, for whoever needs to try this software, they can go over here. Yeah, so they can go over this site. I'm just dragging and dropping it in the chat box. I think I'm not able to share in the chat box.
In chat box, I think you are able to.
I think the chat has been disabled, but I am writing on this particular slide. So this is my contact number. Anyone can contact over here, and I will be sharing the particular link.
You WhatsApp me. I will share on the WhatsApp group as well as on this chat. There is
Yeah, this is the particular trial link site that we have. Anyone can take the screenshot of the particular site.
Any other question from participant side? The session is now open for question hour. Yes, JP. Yes, anyone can ask.
Does anyone have any query?
Is there any real-time application or used by the hospitals of this software?
Yes, sir. So we have Ames as a customer. Then we also have its use case at MGM Healthcare Chennai. So AIMS many departments are using. I do have certain examples of the simulations that I've done for them. So just I need to stop the screen sharing for a minute. I will be sharing it.
Thank you, sir.
I'm just resharing the screen. So So I am sharing my screen. I do have certain examples. So this was an automated segmentation that we had done for one of the patient of AIMS. So we had worked on this particular TMF dataset. And
Your screen is not visible, sir. You are sharing
I
your
think
screen?
yeah, I have shared my screen, sir.
It is visible, sir.
Visible? Okay.
Yes.
So this was one of the dataset that we had worked on. So in this particular, they had to do a CMF planning. This was done at AIMS Neurological Center. Uh, we had worked on it. We had done the automated segmentation. Once this automated segmentation was done, so they had to further work on the particular mandible part. They had to create a implant for the particular mandible data set. So the particular workflow is still in process. We had done the segmentation part, but the particular model generation is still to be done. So this is one of the example data set. Apart from it, we had also done a particular model creation for a cardiac data set. So to use the surgical planning, we had created a model out of same. Let me show that as well. So we had done this particular model creation. Let me share. Yeah. So this model was created to know the internal structure of heart for VSD defect, that is ventricular septal defect. Wherein we had developed a particular four-sectional model. We had connected it using magnets to check for any internal structure. So this is one of our real-world application. So we can use it for virtual surgical planning as well as for the implant positioning and all. And we also have its use case in analysis. So if we need to know the wear and tear of an implant over a period of time, so in that case, our FE analysis can be done, and later on we can check for the simulation results. Thus, we can do further modifications in any implant that we have designed for. So this is one of the four sectional model that we have worked on recently. So that was it from my side. If anyone has any queries, they can kindly ask.
Yes, anyone having any query, please ask. Participant side. If you have any query, then please ask. Okay, over to you, Abhilasha ma'am.
Yes.
Thank you so much, sir, for discussing industry-based applications and introducing simpleware software used in medical image processing in this session. Thank you for sparing your time and be a part of this FDP today. Now, as we move ahead, it is my pleasure to reintroduce our next speaker, Mr. Ravikant Tyagi. We had an exciting session with him earlier also on Wednesday. Today, he will discuss biomedical analysis and applications. Mr. Ravikant Tyagi is a renowned expert and he has 8 years of extensive experience in design, development, and implementation of standalone and client server architecture based enterprise application software. He specializes in Python and data science. He has a broad range of expertise in developing web and desktop applications using MVC and MVT architectures with proficiency in languages and frameworks such as Java and Python Django. For the past 5 years, he has been deeply involved in developing deep learning applications using TensorFlow and PyTorch frameworks. His expertise extends to diverse domains of data science, such as image processing and text processing, with hands on experience on projects like OCR. He possesses strong skills in client side web application development using HTML5, CSS3, JavaScript, jQuery, and Bootstrap. He has a solid foundation in databases, having implemented applications using MySQL, TSQL, and MongoDB, and he is well versed in SQL queries, including joins, nested queries, window functions, group by, sorting, limiting, and regex. His familiarity with JSON-based REST web services and cloud platforms like AWS and Microsoft Azure complements his technical expertise. He has a total 8+ years of experience as data scientist and software professional. He worked as professional trainer in Zebia, co-founder and software trainer at Brain Mentors Private Limited, senior software engineer in HCL, front-end developer in Nestling's WebWorks. He trained 5,000+ students and 1,000+ faculties of colleges like Bennett University, Amity University, Delhi Technical University, IGDTUW, and many more. We are honored to have you with us, sir, and are looking forward to your session. I now invite Mr. Ravikant Tyagi to share his valuable insights with us. Over to you, sir.
Thanks a lot, ma'am. A very good afternoon to all of you, and thanks for inviting me for two sessions, sir. Because for me, one session is not enough, also. So, for this particular session, as we discussed with the participants, that today we'll be talking about GAN implementation, like how we can implement generative modeling. So, that will be today's topic, and we'll be starting with that. So, without wasting any further time, I'll directly move to the code part. Okay. But before that, may I know that you all are familiar with GAN? I think the GAN architecture, you all are familiar. What is generator? What is discriminator? If you're not familiar, I can give you a basic introduction to that. But if you are familiar, you know the basic theory behind GAN, what is a generator, what is a discriminator, then I think we don't have to waste time in the theoretical part. We can directly jump to the coding part.
Actually, all the participants are not from image processing side. Then you can explain some of the basic introduction part and then further.
Okay, okay. So I'll just give you a basic introduction to the GAN. It's not some thing very difficult thing that we have to learn today. Just a second. Right, so I'm going to share my screen. I hope it is visible.
Yes, sir. It's visible.
Okay. You can ping me anytime, whenever you want to ask any question, any query, anything. Okay. So now let's jump to the topic that we have to discuss today, that is GAN. I just want to give you a basic brief about GAN. Actually, I have a complete presentation for GAN, but I don't want to share a PPT. I don't want to, I can share the PPT with you later. I don't want to present PPT here because that will take a lot of time. I just want to show you GAN architecture. Using one single image of GAN, I can explain it to you. You can understand easily what exactly GAN is. Okay. Let's pick any one image like this. Yeah. So this kind of image I'm going to open. All right, now we just have to understand what exactly GAN is. See, GAN consists of three letters GAN, that is Generative Adversarial Network. So now the GAN is like a movie that you might have watched or a web series you might have watched. Can you tell me, have you guys watched a web series Farzi? There was a web series of Shahid Kapoor. Have you watched that web series Farzi? Yes, no, no, no. Okay. Then I have to explain it in my own way. All right, no worries. In that web series, the context is that they generate fake currency. Okay. That is the movie is about fake currency notes generation. So now, how GAN work and how that fake currency generation is going to relate. Let's understand. Now let's say you are an artist, you are like pretty good in drawing, okay? So your art is some great piece of work or something. So you thought, let's generate fake notes. Let's generate fake currency. Your own, like you just want to replicate the exact currency note that India is having. 500 notes, 2000 notes, and 50, 100, whatever it is. Okay. Now, in order to generate the currency note, you'll be doing some analysis. The paper of note is a bit different, the paper that we use. Then what are the languages that are written? What are the sign? What the photo of Gandhi ji? A lot of things are there on the note, correct? So what you will be doing? Initially, you'll start with a random noise. Random noise means you don't have any prerequisite any knowledge about anything. You just start with a random noise. Random noise is nothing, just random dots are there, noise is there. And then there is a real image that exactly is the you can assume it as a currency note. That's the current that the real currency. Okay, your objective is to generate a fake currency, and what you want, how you will validate that this fake note is perfectly generated, that no one can detect. So in that web series, there is a machine that can easily detect if a particular note is fake or original. But in that series, the main objective of the actor is to generate a note that even the machine cannot detect if that particular note is fake or real. Same thing GAN is doing here. GAN objective is to generate some data set that this discriminator, you can imagine that this discriminator is some sort of a detective or machine that is going to detect the input is real or fake. Simple. So now, if you see, there are two deep learning model. One is a generator model, one is a discriminator model. These are simple deep learning models. These are neural network models.
There could be some lag while I'll drop and get onto the train.
Please turn off your mic. Yeah, thanks. So the point is, when we are talking about that, we have two type of data set here, two type of models are here. One data you are generating, one data that you already have. Okay, so there is a real data set that you already have, 0, 1, 2, 3. Let's say these are the images, handwritten digits data you have. And then there is a noise. So basically there are two models. One model is receiving only one input. That is a just noise. Now the generator objective is to generate a fake image. Simple. Generator will be having a deep learning model. You pass some noise, it will return a fake image to you. Okay. Now there is a real image that is being passed to the discriminator. You can see there are two inputs that are being passed to the discriminator. One is a real, one is a fake. So discriminator will tell you if the image that you are passing, if it is a real or if it is a fake. So if I pass a real image to the discriminator, it has to discriminate if it is a real image or fake. Similarly, if I pass fake image to the discriminator, it has to discriminate if it is a fake image or a real image. I hope you now understand the high level understanding of GAN. Everyone got it? You now have the idea what exactly GAN is. So there are two deep learning model. One is a generator, one is a discriminator. You pass a random noise to generator, it will generate a fake image. Then there is a real image that is being passed to the discriminator and also will be passing fake image to the discriminator. So discriminator has to understand if the particular image that you're passing as input is real or fake. That's it. This is the whole idea of GAN. So generator will keep on training the data so that until it becomes perfect, until the discriminator get fooled to understand if it is a real or a fake image. This is the whole idea. So we keep on doing the training part, keep on train the model until the generator generates perfect images. So those images cannot be detected by discriminator if these images are real or fake. This is the idea about GAN. I hope everyone is able to understand what exactly GAN architecture is. Perfect. Everyone, please give me a confirmation. Are you able to understand the high level idea of GAN now you understand what exactly the GAN is? We just need to just simplify the things the way it is, the picture that you are looking here. We just have to do exactly the same thing. Okay, we need a data set, we
Sir,
need a noise, we need a generator model, a discriminator model, and then connect everything step by step. That's it.
sir, how a discriminator knows that it is real or fake?
So basically, you have a real data set that is, let's say, labeled. See, there are two type of images in your deep learning code. You will be having two type of images. One image are real, one image is fake. Simple enough. So how a basic deep learning model work? When you pass a particular image to any deep learning model, if you pass a particular image to any deep learning model, at the end it extract the features from the images and all. Okay, it simply extract the features from the images and compare if it is or you can say we are even going to reconstruct the image so that you can understand that if it looks exactly the real image. So it will be simple, like when you pass a real image to the discriminator, it is going to see that this data you are passing from the original data set or you are passing some fake image. Okay, I'll explain it through the code as well. Okay.
But sir, sometimes we have not real time image or real results which you want to desired. So at that time, I think this type of modeling is not working.
So see, if you have you don't have much images available, you have only 15, 20 or 30, these number of images you have, 20, 30 images you have. Okay. And using that particular data only, I just want to generate other images that looks like the real data set. That is the main thing we have to do. So we can generate n number of images because see, so if you have 20 images, after training on those 20 images, you have generated, let's say 5 or 10 images. Now you have 30 images. Now using 30 images, you can train the model and generate next 10 images. Now you have 40 images and keep on doing that. So
Okay.
this is how you can like keep on generating the fake images.
Hmm.
Okay,
sir.
so
Right,
how
sir.
discriminator will understand if it is a fake image or a real image using the loss. So we'll be having a loss function which will tell the generator whatever the fake image is by if we pass any image to the discriminator, the loss might be too high or the loss might be too low. Okay, so it depends on the loss what exactly discriminator is giving to you. So generator will try to maximize the loss. Real image will try to minimize the loss. So this is the conflict between discriminator and generator that will be going on. So generator will try to fool discriminator, so the loss will be high, but real image will be passed to the discriminator. It will try to understand that the loss should be minimized because we are not doing any classification kind of a problem. It seems to be a classification here, real or fake, but we are not classifying if this image is a dog or this image is a cat. We are not doing that kind of classification. So here we are passing two images and at the end we are going to understand how discriminator understand if it is a real or fake. Okay. So it will simply give you probabilities. Okay. The probability of a particular image and the loss. So there are two things that you will be receiving. This is the maths behind the GAN. If you talk about what is the mathematics behind the GAN, the mathematical functionality is about the probability here. So it gives you the probability of a particular image. Okay. So now I hope we just have a basic high level of understanding. Now what we'll be doing is we are going to jump to the code part, and I'm going to start with a notebook. I received the data from someone from the participants, but that data was like already there are I think 1200 plus images in the data set. Instead of working on that data, what I'll be doing is I'm going to generate one generic can code. You can use that code on any data set. Okay, so that would be a better thing, so that you can simply use this particular code that I'm going to share with you today, and using this code you can utilize this particular code wherever you want, any type of images. So it will be a generic one. Okay, I'm going to use T4 GPU because we'll be having the images data. All right, now which data set I'm going to use, let me show you. So again, you can connect to kaggle.com, and there I found one data set for, let me go to data set. So if we will search bladder, you will get a data set of uh, bladder cancer. Hmm, I think this is the data. Yeah, so this is the data I'm going to use. So there are 4 directories here, center 1, 2, 3, 4, and in these directory we have muscle, known muscle kind of images. So if you want to see the data, how it look, let me show you. So this is the data that already is being available. Okay. And now we want to generate our own fake images based on this data set. All right. Now I hope you all remember how to connect Google Colab with Kaggle.com. I hope you guys remember. And even if you want, you can create your direct one notebook here only. If you want, you can like create your notebooks in the Kaggle.com. Okay, but I generally prefer Google Colab. Not any specific reason, just I prefer. Okay, it's up to you if you directly wants to work with the Kaggle.com. So you can directly create your notebooks here. I will work on the Colab. If you guys wants to work with the Kaggle, you can go ahead. Okay. All right. So if you remember how we connected with last time, I told you about this code. I will copy and paste it here, and just I will use this URL. Copy and replace this, paste, and that's it. Okay. So I don't have to download again the username and the key. I downloaded once, and I can reuse them, and that's it. And now I can easily connect the Kaggle with Google Colab. So I'll be having the dataset with me. If you want to implement with me, you guys can implement as well. But if you just want to understand first, later you can implement. It's up to you. You'll be having the recording, you'll be having the code, everything. All right, so we have a zip file, we have to unzip. So use unzip bladder cancer classification dot zip, execute. Right, now it is done. So if you'll do refresh here, you can see there are 4 directories available to you. Okay, so because there are 4 folders available, center 1, 2, 3, 4, and the same directory we have now here. Perfect. So now this is the first part, that we have the data set with us. Okay. Any question, query, doubts till here? Anything, anyone? Anyone wants to ask anything till this point? No question, nothing. so I can proceed with it. All right. I'm sharing this URL with you all in chat. Okay, in chat, I have shared the URL for the dataset. Now let's get started. Okay, so understand how we are going to implement the GAN. GAN implementation consists of generator model. Generator is a deep learning model. Discriminator, again a deep learning model. So there are two deep learning models we have to create. Okay. Other than that, there will be few functions like load your data set, uh, train your model. So these kind of things will be there. So what are the packages I need? I need TensorFlow as TF. Then I need NumPy as NP. I need matplotlib.pyplot as PLT. I also need from TensorFlow dot keras import layers. So these are the four things right now I need. Okay. I don't want to import all the layers one by one, like conv 1D, conv 2D, transpose, max pooling and all. I just have imported layers and layer inside layer, we have all the other modules available. Now, the very first thing we want to see is how we are going to load the data. See, again, if you remember the last session, in the last session, I told you about how you can simply load and plot the data set. I hope you guys remember that. If not, then please just go to the session and just look at the session once and you will be able to understand. Just for now, I'm showing you how to read an image on port CV2. So you have to write CV2 dot imread. Then let's go to the folder center underscore one slash. Then there is muscle invasive bladder and slash. Let's use one image 0.png. This is the only image I will show you. Okay. I'm just showing for analysis purpose. So we need to read a particular image. Let's see the shape, the dimension of the data. Because we have to be sure like if each image is having the same dimension or they have different dimension, because that is also a good part we have to understand. Okay, so I think all the images might be of same dimension. Now I can try to plot one image to understand how it looks. All right, so this is how a particular image is available. Do plt.access, just set off. All right, so this is the kind of images available to us. I think these are the MRI scan images for bladder. All right, now the objective is to generate the images that looks like these images. Got it, everyone? I hope clear to you guys. If you have any question query, please let me know. Till here, if you have any doubts, you want to clarify anything, let me know. Because after that, I'll be moving to the main implementation part, like how we are going to load the complete data set. Because see, there are like 4 folders are available, 1, 2, 3, 4. How we are going to load these folder? Okay, then if no question are there, no queries are there, then I'll continue. Now let's create a function that will help you to load the data. When you load the data, you can pass like what is the image size you want, you're expecting. Let's say I want to resize all these images into 128 by 128 because see, the by default size of the image is 320 by 320. That might be too big. That might be too like huge size for us as of now. We'll see later if it is required or not. So right now I'm using these, every image will be resized to 128 by 128. Okay. Now let's go to dataset is equals to, I will do tf dot keras dot pre-processing dot image underscore dataset from directory. Now, what exactly I'm doing here is, so in Keras, there are two type of methods available. So when you have images type of data available, in the images type of data, the point is, if you want to load data from the directory, so this is the method. And if you have a data frame, so there is a separate method to load the data from the data frame. Okay, simple enough. Now, in this pre-processing image dataset from directory, you can pass your pre-processing parameters. Like I want to pass here size of the image, color mode. So kind of these things you can easily pass. So the first thing you need to pass is the path. Okay, let's take, it is already giving you hint directory center one something. Okay, let's pass here directory. Okay, directory needs to be the first parameter. So first I'm passing dir. So the first argument you have to pass here is directory. Second argument you can pass is the label mode, because we are using GAN. So in GAN, we are not doing any classification kind of a thing. So you can set label mode as none. It's not a binary classification. It's not a multi-classification problem. So just set the label mode to none. Then what is the image size we are expecting? We have already mentioned the image size in our arguments. Okay, what is the batch size? Shuffle is true. Also, you can do color mode if you want to make sure that color will be always grayscale, because I told you it is going to be a generic code. Okay, see, this is how my data will be loaded. So in the very first thing, we are loading the data set from a particular directory. Now let me tell you how you're going to normalize. So you can use dataset dot map, and I will simply divide all the images by 255. Can anyone tell me what will happen if we divide all the images by 255? Anyone? So this is the function we are going to use.
8-bit image.
Sorry, sir, please come again.
8-bit image.
If we
for
are dividing,
normalization, yeah.
if we are dividing an image by 255, it simply means you're going to like normalize the images between 0 and 1. If I divide it by 127 instead of 255, so it means you are doing with between the -1 to +1. So it depends if you want to divide it by 255 or 127.5. So this is the function that is going to help you to load the data. And I told you everyone, this is a generic code. Remember, it's not specific to the thing that you are doing here. That's why, see, the benefit of today's session will be that you will be learning a generic code. You are not learning a code that is specific to this data. Later, just change whatever the directory name is. Let me show you. See, data is equals to, call this method load data and pass the directory name. Let's say my directory name is center1 and that's it. Simple, that's it. The benefit of this particular code is whatever the directory name is, you can pass it here. The data will be loaded. See, in the center one, it has loaded 641 files. And now let me show you something. In center one, first of all, there are two folders. If I set label mode as binary, it will automatically classify the images into two different categories. But see, because I set label mode is equals to none, so it is going to combine all the images. You can imagine that 100 images are in this folder, 100 images are in this folder. So it means you have 200 images. This code will generate, this code is going to load 200 images. But if you set label mode as binary, so it will load 200 images, but in two different classes. 100 to belongs to one, 100 belongs to second. Are you getting my point, everyone? Is it clear to all? See how easy it is. that you just need to load data, you just need to pass the name of your directory, whatever it is. And this code will work in all the situations. And now if you want to understand what other parameters you can pass, you just need to go to this function and understand the working of this, like what other arguments I can pass in my this particular function. See, color mode is by default RGB. Look at this. So if you see here, color mode is by default RGB. Now I set it to grayscale. By default, the image size is 256 by 256. If you don't pass 256 by 256, so it will take it by default. But I'm saying I want 128 by 128, bed size 32. So these are the kind of default values are available here. You can understand this function in more detail, like what other arguments I can pass. It's like image pre-processing you are doing. I hope it's clear to all. So this is the very first stage, loading the data. Let me comment here. Load the data set. Let's go to the second part. Now, second part, we are going to start to build our generator and discriminator model. The first thing we are going to is build generator model. In the next function, I'll generate another model. Now, if I talk about the generator model is nothing, just a deep learning model, but in a reverse order. Now, what is this reverse order? Let me show you. Let's create a method build generation. Okay, you have to pass the noise dimension initially. Whatever the noise dimension you want, you can pass it here. Let's assume I'm going to pass 100. Initially, my noise dimension will be 100. Okay, let's open that image again. You can open any image. It doesn't matter. We have to open that one only. You can pass any image. You can open any of them, but I usually prefer those images where some data is given. So if I open this image again, you see here, the generator is asking input a noise. Now, what is the dimension of the noise? Anyone can tell me? Guys, can you tell me what will be the dimension of noise?
Noise is an unwanted signal. So
noise will be any dimension. It could be anything. It doesn't matter.
Yeah.
So this particular thing you can understand, okay, that this is noise and the dimension of noise is I am going to take, let's say 100 by 100 or something. But my real image dimension is, suppose it is 128 by 128. So generator now it will generate the image of what size? 128 by 128. Getting my point here, everyone? So noise is I am passing 100. So now what exactly going to happen is you're going to pass a noise that is of 100 dimension to this generator. Generator will implement some convolutional operations and all, and it is going to generate an image of 128 by 128 dimension. Why? Because my real dimension is 128 by 128. See, discriminator is a model that receives the image input. That image input must be having a same dimension, whether it is a fake image or whether it is a real image. Obviously, it has to be same. Got it, everyone? I hope it's clear to all. You can take any number, okay? Just assuming 100 because yeah, generally we start with 100. Already someone in the chat mentioned this thing that standard wise, if you see, you can use 100 or something, some other number. Okay, let's proceed. Now, here comes the most important part, creating the model. So model is equals to ef dot keras dot sequential. Now we have to stack layers one by one. So by default, if we talk about how exactly the convolutional neural network model look like, if you understand the convolutional neural network model, how it look like. So you have convolutional layers, you have max pooling layer, you have then flatten. This is the kind of the convolutional model. But in this particular case, you have to generate that image. So initially I am passing just the noise of 100 dimension. Okay. So suppose I create a dense layer, and then I'm going to say that I want the input shape that will be equals to noise dimension that I'm going to pass. Okay. Now there are few arguments, like if you want to use bias or not, I can mention, no, I don't want to use bias in this case. Set it as false. Now comes the most important thing. That is I want to pass the neurons. Let it be 8 by 256. I'll tell you what exactly this 8 by 8 by 256 is. If you have seen any convolutional neural network model, what exactly it does? When you implement convolutional neural network, how exactly it works? You have an input image, it extract the features from the images, apply the pooling. So eventually at the end, what will be the dimension of the image you'll be having? Obviously, it is going to be reduced. Initially, if my input image is of, suppose, initially, if I say my input image is of 300 by 300, but after implementing convolutional pooling and everything, what will be the dimension of the image? Obviously it will be reduced to some extent, like 16 by 16, or you can say 8 by 8 by number of filter. Let's say you have put it here 64. Are you getting my point here, guys? Do you know this thing? I hope so. Yeah, you know. If no, then please let me know. This is how it works, correct? You have input image of 300 by 300. You pass it to the convolutional after implementing a lot of convolutional steps, after implementing poolings and all. At the end, you'll be having some this kind of dimension, 8 by 8 by 64. Now, 8 by 8 is the size of the image and 64 is the number of filters. Got it, everyone? Please confirm yes or no. That is pretty straightforward thing. Correct. Same thing I'm doing here, because I'm telling you, we are creating a model, but in reverse order. So first thing I'm telling is that this is 8 by 8 by 256. If I multiply this thing, it will become a one single layer. Okay. So now you can implement other operations, like if you want to implement batch normalization, then you want to implement any activation like leaky ReLU or ReLU something, and then you're going to reshape it. So why we reshape? I can mention here, reshape the output into a 3D tensor. Now, why 3D? Image format. See, just connect the things. I told you that we have 300 by 300 image. After implementing CNN, you have 8 by 8 by 64. Now, what is the next step after this? Let's say now my CNN is implemented. What is the next step? Next step is flatten. Next step is flatten. Now, what flatten does? Anyone? What flatten does in this particular scenario? Anyone? Any answer here? What flatten is going to do in this particular scenario? In CNN, what flatten does?
Reduces dimension in one dimension, reduces
Exactly.
data.
So it will become 64 into 64, whatever the square of 64 is. Okay. So at the end, that particular number of neurons are there. This is what exactly it do. Isn't it same thing we are doing, but in the reverse order? Isn't it the same thing we are doing in reverse order? So at the end, I'll be having 8 by 8 by 256. I'm assuming this. Might be it will be something else, but if I am assuming this parameter, so in the generator model and in the discriminator model, I'll keep the same dimensions. So I'm assuming this is the last layer. Now, if this is the last layer, what will be the layer before flatten? Before flatten, this might be the shape, correct? Yes or no? Obviously, yes. If this is going to be my last layer, then this is going to be my second last layer. Obviously, it was in this particular shape, 8 by 8 by 256. So when I apply flatten, it becomes 8 into 8 into 256. Correct? I hope everyone is able to understand. Now comes automatically it is giving you suggestions. See, obviously we have to do this thing, but let me show you how. So layers dot conv 2D transpose. Now comes this conv 2D transpose. So by default, we do upsampling and downsampling. There are two things. So here we need to do upsampling. By default, we do downsampling. In convolutional neural network here, this is known as downsampling because you are downsampling the images, but you are generating the image. In generation, you are upsampling the image. Okay. So here we are applying upsampling. So we upsample it to 16 by 16. By default, it is 8 by 8 here. So I will upsample this image to 16 by 16. How? Just pass strides as 2 by 2. And how many filters you want? Let's say 128. I was having 256, but now I want 58 by 58 and then comma. I need a filter window size of kernel size of 5 by 5. Strides value will be 2 by 2 and you can keep padding as same. Okay. And then mention use bias false. All right. Now again, batch normalization will be there and leaky ReLU will be there. Okay. Now upsample it to 32 by 32. Perfect. Same thing we have to do. So you can just copy this and paste. So upsample it to 32 by 32. So you can mention here, let's say we are going in reverse. So it will be 64. It will be 5 by 5. Everything will be same. Don't have to change anything. Okay. So it has reached to 64 by 64. Now you can upsample it further. Just copy and paste. Upsample it to 64 by 64. So it will be 32. And what else I need to do? 5 by 5. So we can do here. Torus shape is there. Then 1, 2. We don't have to upsample if I take, because we have taken 128 by 128. Okay, let's keep it 64 because it will be very slow if I keep it as 128 by 128. It will take a lot of time for 128 by 128. So let's keep it till here. So now I can show you that 64 is the number of filters I have, and then I don't have to upsample it further. Now I can do final transpose. Okay, so it will be upsample into 64 into 64 layers dot conv 2D transpose 1 comma 5 by 5 comma strides 2 by 2 padding same. Now what is the activation you want to implement? Let's implement 10H and all. Okay, perfect. And then return the model. That's it. Look at this generator code. This is the generator model code. So guys, please look at the code and let me know if you are able to understand this code or not. So this is the generator model code. So this is CNN in reverse order because we are not extracting the feature from the images. We are generating the image. So that's why we are doing it in a reverse order. So this is how generator model is implemented. Now we have to implement the discriminator model. Can we proceed, everyone? Is it clear to all? So I can proceed to discriminator model. Please give me a confirmation, everyone. Yes, we can proceed. All right. So now once you have generator model with you, now we can implement the discriminator model. You don't have to pass anything to this. Now in discriminator model, we need sequential and then obviously the first layer will be 64 by 64 by 1. Okay, why? Because our data dimension is 64 by 64 and also we are generating a 64 by 64 by 1 image here. That's why. So we can directly implement this code const2d. Now implement a leaky relu on it. Drop out, you can mention if it is up to you. Then again, now we are doing downsampling. You can say now the downsampling part is going on. So in the very first part, we are downsampling. So here I will write a comment for you, downsample it to 32 by 32. Okay. Then downsample to 16 by 16. Okay. And then downsample to 8 by 8. Just copy this. So 64, 128, 256. No. If I do, okay, 8 by 8, 256, 5 by 5, stride padding, and then I will do layer dot flatten. So flatten will simply flatten the complete network and then dense 1. That's it. So we are done with the discriminator and generator model. Both the models are implemented here. See, if you have any issue or anything, please let me know. Now, if you see discriminator and generator exactly opposite to each other, look at this. So you start with the downsample part of 32 by 32, 16 by 16, then 8 by 8. Correct? What we are doing here, we are expecting 8 by 8 into 256, then 16 by 16, then 32. Isn't it same? And we are done with discriminator and generator model both. So everyone, are we clear? That now we have generator and discriminator, both the models.
Papa.
Yeah, mute.
All right, guys. So if we are clear, then I can proceed. So now finally you have both the things available, generator and discriminator. Now, in order to test, like if you want to simply understand how it is going to work and all, you can generate a random noise, pass it to the generator. It will generate some fake image. Then that particular image will be passed to the discriminator model. Okay. So you can consider it like if I have to implement that, I can mention. Let's take noise dimension as 100. Then generator is equals to build generator noise dimension. I'm not using discriminator, just generator. So we are testing our generator model. Okay, now generator model is tested. How I'm saying it is tested? Because see, if I call build generator and I pass 100 as a dimension, it generates the image. Now in a generator variable, you can see a lot of things. First of all, if you want to see the summary of the model, okay, you can see that this is the summary of the model. This is how my model looks like. See, similarly, you can generate the summary of the discriminator model. It will tell you in detail how many layers are there, because I have seen in a lot of research work when you are doing it for the PhD and all, every time you have to mention the layers, the shape and everything detail about the model. So it's not very difficult. You just need to mention, just go to the summary method. Okay, it will print the complete summary of the model for you. All right, let me comment it. Now, once the summary is generated, I'm saying that it has generated some image for us as well. Okay. And for that, I can do one thing, generator. I can do generator if it is. Predict. Let me check the output. Image. It is going to be a fake image. Fake image dot shape. Okay, then plt dot I am show fake image, and then let me show you the output, the fake image that it has generated. See, this is the output after implementing generator model. Now you can ask like why this look like this, because we have not trained the model yet. So now you can understand, okay, once we start the training part, it will keep on generating the images and it will keep on improvising the things. Getting my point, everyone? Till here, I have executed the generator. I have implemented the build generator function. Now we pass a random noise to it. It generates the image, fake image. Then I resize the fake image. And finally, after resizing it, I can see this is the output. This is the fake image that generator has generated. Got it, everyone? So once we have the fake image generated with us, now we can continue with the rest of the things. Like we have discriminator and all. Now we have to continue with the model part. Any question, queries, doubts, anything till here? Anyone?
Sir, can you please send your collab notebook link?
Yes, ma'am, I'll share.